So good afternoon, Professor Rawat, Professor Hitendra M, and also good afternoon, Professor Melati. I hope that you are doing well.
Good morning.
So good afternoon, Professor. No, not good afternoon. Good morning. I'm sorry. Good morning, Professor Commeiras. I hope that you are doing well. Okay. How's it going in India, Professor Rawat and Professor Patel? No? Okay. Uh, 15th. Professor Melati.
Ya, Assalamualaikum.
Waalaikumsalam.
Kabar?
Apa kabar? Baik-baik. Bagaimana Kak Penang?
Alhamdulillah. Itulah masih ada fiction lagi lah. Walaupun reda sedikit. Tapi masih itulah. Banyak SOP.
Iya, sudahlah. Prof Rahana sehat ke? Prof Rose, Dr. Rose.
Sehat. Ya, sehat.
Sehat, alhamdulillah. Profesor Rawat. I think that is still in mute. Hi. It is very nice to meet you in here, finally. Yeah. Professor Patel.
Good morning.
Good morning. Yeah. How's it going in India?
Yeah.
Good.
Everything is fine in India.
Good. Nice to hear that.
Yeah, thank you.
See that. Admit. So I think that we can start at 1:05 or 1:10, I think, yeah. Professor Rawat, I'm afraid that I can hear your voice.
Mungkin perlu unmute. He is in the meeting.
Okay.
Yeah, so he will join second talk.
Okay, that will be fine.
Yeah. Maybe Idham.
Yes.
You should write something to him.
I'm sorry?
Write in the chat for him, because I think something with the audio.
Yeah.
Something wrong with the audio. Maybe you can write to him in the chat.
Yeah.
Because he wrote just now. Oh, he can hear us.
20. Oke, Mbak Joan. Mbak Joan.
Yes, sir.
Okay, maybe you can start.
Okay, thank you.
Okay.
First of all, may I confirm if my voice is clear enough for today's event? Hello?
Yes.
Yes, yes.
Okay. Greetings to everyone, wherever you are. Good afternoon to our distinguished speakers, Professor Melati Khairuddean, Professor Diwan S. Rawat, Professor Hitendra M. Patel, and Dr. Idham Darussalam, as well as to all the audience that are present here today. I hope all of you are in good health. Welcome to program of Guest Lecture Series, World Class Professor Program. Allow me to introduce myself. My name is Joanne, and I am honored to be your MC and moderator for today's event. Before we start, I will be reading the code of conduct for today's event. Code of Conduct, Guest Lecture, World Class Professor 2021. Rules for the guest lecture today. Number 1, participants must use their real names, not pseudonyms or calls, by following the format participant _ name, moderator _ name, host or committee _ name, or speaker _ name. Number 2, participants please wait for approval from the host in order to enter the meeting room. Number 3, when entering the Zoom meeting, participants and related parties are not allowed to turn on the audio microphone before being invited by the moderator and can be turned off directly by the host if it is deemed quite disturbing. Number 4, when entering a Zoom meeting, participants and related parties must turn on the video. It is allowed to turn off the video if there are signal or network problems or it interferes with the smooth use of the Zoom meeting. So please do turn on your respective video cameras as a show of respect for our distinguished speakers of today. Participants can use the chat room to interact with fellow participants for the needs of the event by using polite and courteous language. Number 6, participants are not allowed to say rude and disrespectful things. And number 7, participants must follow the applicable rules for today's event. Okay, now I would like to humbly ask all of our participants to fill in the presence form from the link sent by our committee in the chat room. All right. To officially start today's guest lecture, I would like to invite Dr. Idham from Universitas Gadjah Mada to give the opening remarks. Good afternoon, Dr. Idham.
Okay, good afternoon, Mbak Joan.
The time is all yours.
Okay, thank you very much. Okay, please allow me to share my screen. So can you see my screen?
Still loading, sir.
Okay.
Yes.
Okay, good. So while waiting for the other participants, please allow me to introduce you to our program. Okay, good afternoon, good morning, Assalamualaikum warahmatullahi wabarakatuh, Om Swastiastu, Salam Kebajikan Nama Budaya. Okay, I hope that you are all doing well. Yeah, thank you very much for your time for attending this guest lecture. Yeah, for information, the guest lecture is part of a program of world-class professor program. So this program is an annual program. Yeah. Conducted organized by the General Directorate of Higher Education from the Ministry of Education, Culture, Research and Technology, Republic of Indonesia. So in this year, yeah, we would like to conduct a world-class professor program in the theme of the green chemistry approaches for efficient synthesis of nitrogen heterocyclic compound. Yeah, so in this program, yeah, is organized, jointly organized by two universities from Indonesia and also from France. Yeah, from Indonesia, it is me, yeah, Dr. Muhammad Idham Darussalam Marjan from Universitas Gadjah Mada, and from France, yeah, the invited world-class professor is Professor Commeiras, yeah, from Aix-Marseille University in France. So please allow me to show to say about the objectives of this program. Yeah, the main objectives of this program is to improve and strengthen the international network, yeah, through world-class professor program, yeah, between our university, Universitas Gadjah Mada, and with other reputable university in abroad. In this case, with the Aix-Marseille University in France. Yeah, and then we would like to improve the research quality. Yeah, especially in the field of organic chemistry, because me and Professor Commeiras are working in the organic chemistry, and then we would like also to increase the capacity, yeah, insight and also skills of the Universitas Gadjah Mada academic community, yeah, the student and also the lecturer and the researcher, especially in the field of organic chemistry. And then we would like to improve the quality and the quantity as well of our scientific publication. Yeah, and also at least at the end, yeah, we would like to increase the ranking of our university in the world university rank. Yeah, so these are the objective of our program. So to do so, to realize our objective, yeah, we would like to perform several activities in our world-class professor program. Yeah, there are two programs, yeah, including the fine-tuning publication and also the guest lecture that we have today. Yeah. So about the schedule, yeah, we have already conducted several activities. Yeah, we have completed the fine-tuning publication. There were 7 sessions. Yeah, and we have already conducted the first guest lecture. It was at the 1st October 2021, and now we have arrived in the second guest lecture. Yeah, at 29 October 2021. Yeah, we still have 2 more guest lecture 3 and 4 at 12 and 19 November 2021. Yeah, please save the date. Yeah, and then about the fine-tuning publication, we have several speakers in here. For example, Professor Commeiras and me in here in the middle, and also Professor Rawat, yeah, from Indonesia. Now about the guest lecture, as I told you before, we have 5 series of guest lecture, and today we are in the guest lecture number 2, yeah, in here. And allow me to introduce about our speakers. Yeah, in the first guest lecture, we have Professor Black, yeah, from University of New South Wales, Australia, and then we have Professor Hayashi from Tohoku University, Japan. And now it is our honor to have Professor Khairuddean from University Sains of Malaysia, and then Professor Rawat from the University of University of Delhi in India, and also Professor Patel from Sardar Patel University. The third guest lecture, we will have Professor Commeiras as our speaker, as well as Professor Cravotto from the Università degli Studi di Torino, Italy, and also Professor Gadjah, yeah, from the Universities of Mont Blanc in France. The last one, we also have Professor Commeiras as our speaker, and also we will have Professor Panik from the Prince Muhammad bin Fahd University, Saudi Arabia, and also Dr. Valeria Palermo from Universidad Nacional de La Plata, Argentina. Yeah. So for your information, yeah, this event, yeah, we have the participants from the students, researcher, lecturer, teacher, as well as employees, yeah, from university, research institute, senior high school, even the senior high school, and industry and non-government organization. Yeah, coming from Indonesia, Japan, Nigeria, China, Pakistan, and Thailand. And today we have at least 117 participants who registered in our second guest lecture. So the next one, why? Okay, please allow me to introduce our world-class professor, yeah, which is Professor Laurent Commeiras. Yeah, now he is a professor from Aix-Marseille University in France. Yeah, so now he did PhD education from 1999 until 2002, and then now he is responsible for the head head of the Master of Chemistry in Aix-Marseille University, and then his work of interest, yeah, in the development of new methodology, including the formation of polycyclic complex structure, as well as the total synthesis of natural products and the molecular of interest. Yeah. Okay. So without further ado, yeah, I wish you. I hope that you will have a good lecture today. So please enjoy our guest lecture. So I will talk about a bit about our lecture today. So today we will talk about the efficient synthesis of the heterocyclic compound. In this case, we would like to employ the ionic liquid as the media of the synthesis, and then how to design the pharmaceutically active compound, yeah, by using the hybrid approach, yeah, in here. And also we will talk about the chemistry and also the biology of the pyrazolines derivatives. So without further ado, please enjoy our guest lecture. Thank you, Mbak Joan. Yeah, terima kasih and merci beaucoup.
Thank you very much for the warm welcome, Pak Idham, Dr. Idham, and also for all of our professors. So another thing, while we are still fresh, so let us all take a picture beforehand. There are a number of slides. Oh, only the first slide have their cameras on. So for the other participants who would like to join for our photo session right now, can you please turn on your cameras for a second, and I will be taking a picture, okay? So 3, 2, 1, smile. Okay, that is all for the documentation today. Let us move on to our first speaker. Allow me to introduce our first speaker of the day, Professor Melati Khairuddean from Universiti Sains, Malaysia. Professor Khairuddean received her PhD in organic chemistry from Kent State University, Ohio in 2009, and her field of specialization is organic chemistry, including synthesis of natural products or liquid crystals. Since 2009, she has a number of both completed and ongoing research grants in the fields of organic chemistry about synthesis and characterization of new compounds, such as cyclo-3-phosphazine derivatives and their fire retardant properties, new compounds with chalcone and heterocyclic scaffolds and their biological activity studies as well, and natural products of chemistry, such as phytochemical and biological activity studies of pure compounds from plants, as well as modification work on some identifier bioactive molecule isolated from natural products. Professor Khairuddean has up to 83 publications and counting. She has received a number of appointments or achievements, namely Timbalan Dekan Akademi Gadjah dan Antara Bangsa, and Anugerah Perkhidmatan Cemerlang in the years of 2011 and 2017. And she is also not new to conferences and symposiums where Professor Khairuddean has attended as guest lecturer and visiting professor in universities such as Universitas Negeri Yogyakarta and Universitas Pendidikan Indonesia. Wow, now that we all have heard the CV of our first lecturer, surely Professor Khairuddean's achievements makes it the more interesting for all of us, and we cannot wait to learn more from such an amazing professor. So let us all take off our capes and empty our glasses. Let us get ready to learn and fill our glass with new knowledge. Let us give a warm welcome for Professor Melati Khairuddean. Good afternoon, Professor. How are you today? Hello.
I am good.
Oh, that's happy to hear.
Thank you, Joanne.
Yes, you're welcome. So for the next 40 minutes, the time is yours, Professor, and I will be sending a reminder through the private chat once 30 minutes has elapsed and there are 10 minutes left. The time is all yours, Professor.
All right. Okay. Thank you, Joanne, for such a kind introduction. Can I share the slide? Okay. Oh, yeah. Okay. All right. All right. Okay, can you see this? Can you see the slide?
Yes, ma'am.
Okay, thank you. All right, first of all, Assalamualaikum to all the participants. Good afternoon and good morning to those who are in different part of the world. Okay, thank you again, Ms. Joanne, for kind introduction. And thank you, Dr. Idham, for inviting me to be one of the speakers in this program. Okay, I'm really very honored to be here and to share some of the ongoing research work in my group with all of you. Actually, there's nothing so special about the work. It's just some little bit interesting because it is chemistry and combine chemistry with some of other new knowledge from other fields. Okay, so this is the title given to me, and before I start, let me introduce a little bit about University Science Malaysia, where I come from. Okay. All right. So this is University Science Malaysia. This is the top view of the front entrance of USM. USM is University Science Malaysia. Okay. So this is the university vision, transforming higher education for a sustainable tomorrow. Right. And then you can see this Penang Bridge that connects the island to the mainland. Right. So this is actually, we are on the island, a small island. All right. So all right, this is the map of Penang, just to show you where I am. Okay, so this is the whole Malaysia. So this is the island. This is the map of Penang Island, where the main campus of USM. Okay, it is located. Okay, just across the bridge here. Okay, so people from mainland can come to Penang through two bridges. The first one is right in front of USM. Okay, so this is the first one, right in front of USM, and then the second one is a bit is much longer bridge and a little bit further away, closer to Penang International Airport. So here it is. Okay, so this is the second one. So, and then this is Malaysia, and USM also have branches all over Malaysia, right? The engineering campus, and then the health campus, and then the dental institute, management center, etc. Okay, so this is USM, also known as University in the Garden. Okay, so, and this is School of Chemical Sciences. Right. And okay, this is the School of Chemical Science inside there. Okay, and this is me. Okay, this is me, and all these some details about me and some contact. And this is the event. Okay, I want to thank again Dr. Idham for the invitation. And back to the title. Okay, pyrazoline, synthesis of bioactive compounds as anti-cancer agents. So the biological activities would be the anti-cancer. One of the target scaffold is the heterocyclic ring. So why are we choosing heterocyclic compounds? It is a key structural component of many anti-cancer drugs in the market. So almost 2/3 of it. So they play a vital role in metabolism of the living things being utilized here and there, varieties of intermolecular interactions that allow these compounds, such compounds, to bind with the enzyme. So it shows very good interaction. So, and also it is very easily synthesized. If it is not, then I will not do it, because I don't want to be, wasting the time and also a lot of money to buy chemicals. Now chemicals are not cheap, right? So if it is easy and well, you can get good yield of compounds, then why not, right? Okay, so we are interested in looking at the biological activity anti-cancer. To look at the cancer. What is cancer? Just a little bit. It is about a transformation of normal cell become abnormal cell. It will divide without control and spread to the nearby cells to form a tumor. So this cancer, the treatment depends on the type of cancer and stages of the diagnosis. So not all the cancer treatment is 100% effective for most of the tumor types. So why do we need to develop new anti-cancer agents? Since there are already a lot, but because of the drug resistance, low specificity, and also high toxicity of the chemotherapeutic agents, so these all results in very serious side effects. Okay, so that is why we try to, have this thing is now having this continuous, searching for a new type of anti-cancer agents. So our focus is on the heterocyclic compounds, which has a ring containing at least one element of other than carbon. That is the heterocyclic ring. In particular, we are talking about pyrazoline. This heterocyclic ring has this nitrogen atom inside the ring. Okay, so this is the pyrazoline, 5-membered heterocyclic ring. There are 2 adjacent nitrogen, 3 types of pyrazoline. There are also 1 pyrazoline, 2 and 3 pyrazoline with different position of the 1 endocyclic double bond. So it can be found in varieties of the leading drugs. So 2-pyrazoline is the most studied. So what we are doing, we are focusing on these 2-pyrazoline compounds, right? And there are also several drugs in the market with pyrazoline ring. These are some of the examples. Okay, so what we do in the research work is first we try to design new compounds that have this pyrazoline ring. And the most common method to synthesize this pyrazoline is from the chalcone compounds. So that is why we synthesize the chalcone and then use this chalcone to make a lot of heterocyclic ring compounds. Because from chalcone, you can get these pyrazoline compounds, and you can also get other heterocyclic ring compounds from chalcone, right? And then to understand the properties through molecular docking. So we just do a little bit here and there about this molecular docking, and then we get all these values, okay, and try to meet these values with the Lipinski's rule. Right? And then we send to our collaborators for the cytotoxicity study and to understand the structure-activity relationship. Okay, so one time doing this, that means you are not going to get very, very good compounds. Is okay. We can always do modification on the compounds and try to get better one. Okay, so these are the common, the general synthesis that we did. Okay, the type 1 is the monopyrazoline. Okay, we get this monopyrazoline type. We have only one pyrazoline in the compound. So the second type is the bispyrazoline, whereby you have two sides of chalcone, and this chalcone transform into two pyrazoline. So that means we have the bispyrazoline type. And the third one is where you have a heterocyclic ring that have this so-called chalcone, right? But then it can form the fuse type pyrazoline. Okay, so that will be the type 3 pyrazoline. Right, this is how we numbered it, so that we don't get confused, because after a while, when you get a lot of data, then you will get confused with that. Okay, so in drug development, a lead compound is optimized by modifying or add different functional groups. Okay, so we have this structure activity relationship and try to understand, relying on the binding affinity or some of the parameters, then we make the compounds better. Okay, we do molecular docking, performed to predict both the type of interactions and also the binding energies of the ligand. Okay, so the binding energy of the target for its of a drug, that means our compound, for its target, right? That is the enzyme. If binding energy is low, then it is good. Okay, you will get stronger and more stable bond, right? So for this molecular docking, you have to do several things. That means to prepare the protein, prepare the ligand, and also then only you get to have the values by using Autodoc 4.2. Okay, so there are a series of things that finally you will get some of the values from there. Okay. Then the cytotoxicity study, that is the biological activity that we are working with our collaborators. Okay, we are interested in looking at this. So we work with a few collaborators, and also we have also a collaborator in India that helped us to do this anti-malaria. Okay, not anti-cancer but anti-malaria. So it's like a going away from what we are doing. Okay, so it's still a biological activity study, how to relate to the compounds. Okay, so and then the effect of compounds against chosen breast cancer. Okay, we are focusing on breast cancer, even though it is cancer. There's a lot of different type of cancer. So we are focusing on this breast cancer. So chosen breast cancer cell lines, then being measured, and then being classified, okay, based on the values given, the IC50 and all that. Then we get also the inhibition constant values and all that is from the docking. And then we get the selectivity index from the cytotoxicity values. Okay. So those are the values that we will get after several series of assays and docking, molecular docking work. All right. And various molecules have been studied, whether they bind well with estrogen receptor alpha. Okay, that is a very crucial receptor for breast cancer, plays important role in cell proliferation. Okay, usually in our case, we will choose the MCF7. Okay, that is the ER positive, right? And then also the MDA-MB231. And also another one is the normal breast cell, MCF10A. So all our collaborator will do for us. We will not go into it, but we are there. We are there together with them. Okay, looking but not doing, right? Okay, so the normal breast cell is the positive control to determine the selectivity index. Okay, and continuous research on designing of new molecules are needed, definitely are needed. Okay, so this is the first one. Okay, this is the pyrazoline ring. This is the compound. All right, so this is the compound. So what we did, this is the nitro substituent. We put the electron withdrawing group, nitro substituent, to explore the potential of anti-cancer activities. Okay, and also some of the electron withdrawing group and also electron donating. Okay, and R is actually all those substituent attached to this pyrazoline ring. Okay, so this is the pyrazoline ring. So R is all those substituent. Okay, and then we have managed to synthesize all the compounds. And these are the cytotoxicities values that we get. Okay, we find that, okay, looking at 1A, shows just moderate. 1C is okay, better. After 24 hours, we get about 6.5. And then with the selectivity very high. Okay. All right, so this type of compounds. And then looking at this, we have pyrazoline compounds. We evaluated against breast cancer cell lines, MCF7 and MDA-MB231, 2 type. Okay. And also normal breast cell lines, MCF10A. And then pyrazoline 1C shows moderate IC50 values when exposed to MCF7 for 24 hours. Right? So so compound 1C is used in the discussion. So we find that nitro group that is attached at the para position of the benzene ring will show one oxygen of the nitro group will form a hydrogen bond. Okay, so this is the from the docking. Okay, we can see that this is a nitro group. Okay, one of the oxygen is attached to this. They form a hydrogen bond with the amino acids. This, right, as observed in the molecular docking. So this hydrogen bond could improve the binding interactions of the ligand receptor. So pyrazoline 3D with electron donating substituents shows lower binding energy. Okay, so we can go back and see. Okay, 1C just now looks promising. Okay, 1C with very high selectivity. Selectivity should be more than 2. Then it is considered very good. So then pyrazoline 3D. Okay, 3D is with the ethoxy group, the electron donating. Okay, but then the values are not that good. But the binding energy looks good, right? So, however, better cytotoxicity activity against MCF7 was observed for 1C compared to 3D, even though their binding energy are comparable. Okay. So those are the things, some of the things that I take out from the paper, right, from the publication. Okay, so this is the research number 2. Okay, we try to form a hybrid compounds. That means they have 2 or more known scaffold. Okay, to give compounds with promising pharmacological potential. Okay, due to the sum of each of the scaffold potency. So that means each scaffold has their own potential. So sum of it. So hopefully it will give promising activities. So these hybrid compounds can simultaneously also target more than one mechanism. So it enhance the activity or selectivity and also reduce the adverse drug effect. So we have here, this is the okay. My mouse cannot work. Sorry, I cannot show. Okay. Okay. All right. Okay, now I can see the mouse. Okay. All right. Okay. So we have pyrimidine ring, and then also some of the aromatic heteroaromatic ring. Those are the rings here. Those are the rings here that will attach to here. So we have synthesized all these compounds. Okay, and managed to get all these compounds listed here. Okay, we find that cytotoxic activity of pyrazoline derivatives against breast cancer cell lines reveal that 4A is the best. I mean, 4A, 4A is the best proposed candidate against hormonal breast cancer. Okay, so hormonal breast cancer is this, MCF7. Non-hormonal breast cancer is the MDA MB231. That will be for 1C. Okay, 51 is a bit too high. It is just a moderate thing. Okay, and further investigation and possible modification will be done to improve the activities. Okay, the values are not that great. And we have that binding energy of those compounds. Okay, even though this 4A, we cannot really discuss on this binding energy because it is not that great, negative 7.66. It should be like negative 10. Then it is considered very good. Okay. Right. Then another part is the mono, the type 1 also is the monopyrazoline compounds, whereby we have only one. Mono means one of the pyrazoline ring. And now we're trying to put other things. We are going to put a naphthalene ring here. Okay, this is to increase the hydrophobic surface area. So put bulkier group, such as naphthalene. And here we have pyrazine, the 6-membered nitrogen-based ring with pi donor properties as additional scaffold. This is to increase the binding affinity. So these are all the compounds. We have synthesized all this. And then we got all this 1A to D and 2A to D. Okay, one is with the naphthalene 1 position. The other one is the naphthalene 2 position. All right, so we find that. Okay, to increase binding affinity, what we do, we can add more groups with pi donor properties to form hybrid molecules. Okay, so increase hydrophobic surface area of the benzyl end of the ligand. So we replace that with bulkier group, such as naphthalene. So naphthalene ring has more number pi bonds with more resonance structures and more delocalization. So compounds hopefully will be more stable, right? Okay, and then the position of the naphthalene ring does not really give significant difference. Maybe it's just 2, we cannot really see. Maybe in the future we will have maybe even substituent on the naphthalene ring. Okay, so this is the 1B compound. Okay. All right, so here we have pyrazoline 1B shows distinct interactions. Strong hydrogen bond and other hydrophobic interactions increase, we can see that increase the binding affinity. So as we have said just now, we can have a bulkier group and also add more of the scaffold with pi donor properties. Yeah. And then it will form hybrid compounds with 2 or more known scaffold to give compounds with promising activities because of the sum of each of the scaffold potency. So it can also target more than one mechanism. We are into there, try to collaborate with another group, trying to learn about the mechanism of the compounds. So it would take a while. Okay, all right. And then another work is this type of type 1. We are still in type 1. Okay, so making these pyrazoline compounds having a thiophene ring. Okay, so hybrid that with thiophene ring. So we have the pyrazoline, so now the thiophene with a chloro atom or with the dichloro atom. Okay, so we have this, and it shows potential anti-cancer activities. So all these compounds, okay, all these compounds have been synthesized and characterized. Okay, so we have all these compounds, and a new series of mono, okay, it's a monotype, monotype compound. So mono because there's one pyrazoline. So we have a thiophene pyrazoline hybrid compounds that shows acceptable binding energies. Very good, all very good. All about -11. So that is very good. So, and also the values for Ki is very low. The best values of binding energies, we find that it is the 3E, for 3E and also 7E. Okay, and further evaluation of these compounds against MCF7 breast cancer cell lines and also normal cells will give the IC50 values and selectivity index. Okay, we are still waiting for the results. So these are the molecular docking of the compounds that shows very low, very good binding energy. So we have all these interactions for both. This is the 3E and also 7E. Okay, and another one is that's the type 2. Type 2 is the bispyrazoline. Okay, we have the bispyrazoline. That means we have two pyrazoline there. Two of their pyrazoline. So that actually comes from two of the chalcone. Okay, so the same thing, we also have this thiophene, one chloro, and the other one is two chloro atom. Okay, so we have synthesized all these compounds. So we hope this one will show potential anti-cancer activity. Because just now, the previous one is only one part of it. Now it's like you double the ring, the pyrazoline ring. Okay, so we did molecular docking. The value shows binding energies of pyrazoline 1A. Okay, looking at this, we did the molecular docking for all the compounds that is intended. So we did all the docking, but only select the good one. So pyrazoline 1A shows binding energy to the estrogen receptor alpha. That is the best. Okay, 1A shows the best. And then we have this. It also predicts the best structure with the highest affinity to bind to the breast cancer cell lines. So other pyrazoline compounds also show good binding energies. So all the Ki values are low. This is good. And also others is also good. Okay, 2E is also good. Okay, so we chose one, and to show it here, the interactions. We have the hydrogen bonding and also some of the hydrophobic interactions. Okay, we find that for this compound, this type of compound, the bispyrazoline, they have larger structure. So they have increasing number of bonds. So more interaction with the residue of the amino acids in the active site. And this could have enhanced the anti-tumor activities. And also the substituent of one or two chloroatoms does not show significant difference. So that means 1 and 2, they are more or less the same. Okay, they did not show any, it's significant. They are not significant. So further analysis is carried out in vitro to test the activity against some selected breast cancer cells and also the normal breast cells. Okay, so now pending, a lot of our compounds are pending due to the pandemic situation, and our collaborators have problem to go to the lab because they are dealing with this cell culture and all that. So most of the labs, we cannot go to the lab. Then we also cannot get any of the results at the moment. Okay, and another one is the fuse. Okay, so this is the fuse compounds. Fuse pyrazoline. Okay, fuse together. That means we have the core structure of the cyclopentane or cyclohexane, right? And then with one of the double bond here or there, then we will form the fuse type pyrazoline. Okay, we have synthesized all that and get this. Right, so these are the, sorry. Okay. Right, so these are the four, okay, the four compounds being synthesized. Okay, so these fused pyrazoline compounds were evaluated against breast cancer cell lines, MCF7, and also normal cell. So we find that 1A and 4A, they were more potent against MCF7. So selectivity is very high, which is good. Okay, making them maybe a potential candidates to further study their structure. Okay, maybe do some of the modification on the structure. Right, so this is the fused pyrazoline. Now we have the cyclopentane system. Cyclopentane 1A and 2A. Cyclohexane is 3A and 4A. Okay, so for this cyclopentane system, chlorine of fluorine substituents at the O2 position of the aromatic ring, they show very good cytotoxic activity. Okay, so here, so these are the position. Okay, so you put chlorine or you put fluorine for both. It has to be for both. Okay, so 1A is better in terms of activity and selectivity. That means when you put Cl, it's better than you put fluorine. Right. So in the infused pyrazoline cyclohexane system, okay, cyclohexane we have 3A and 4A. Then the fluorine substituent in compound 4A showed best activity and selectivity, while compound 3A did not. Okay, so compound 4A is better than 1A. So this one could be promising type of compounds for us to do modification. Okay, so just now we have this, we can see that, okay, this is the cyclopentane, this is also cyclopentane, not that good, this one okay. This one and this one looks okay. This one looks like inactive, not that good lah. So for this, if you have a cyclohexane, then putting fluorine is better. So we are still trying to understand and have and do some of the other modification to understand why it behaves like that. All right. Okay, then this is the other type, where we have this instead of electron withdrawing, just now is electron withdrawing, but now we have here is the electron donating, is the ethoxy group. Have the ethoxy group, put it there, and then we get this compound. Also the same, the cyclopentane and also the cyclohexane. So we get the series of compounds. So these are the series of compounds, and we tested. They were evaluated. We get the results. This one happened in between MCO, very early time. Okay, because they were working on that before it went haywire. Around 2020, early 2020. Okay. So now these compounds, it was evaluated. So we have MCF7, MDA, and B231, and it means with receptor and without receptor, and also the normal cell. Okay, we have normal cell. Then we can do the selectivity, get the selectivity index here. Okay, it seems that we find this pyrazoline 1A and 2A. Okay, so this is 1A, this is 2A. They are all the same. It's just that one is the cyclopentane, one is the cyclohexane with this thiosemicarbazide, you know, the substituent there. Okay, so they give this, okay, looks okay, even though it is more than 10, huh? Should be below than 10 would be better lah. Huh? So we get around there, and then it shows that both they prefer inhibition towards this MDA MB 231 instead of MCF7. So, and then the selectivity is also, this is after 24 hours. So looking at after 24 hours, you get this. Selectivity is around going to 4. And here also, we get this. And this is also a bit similar. Okay, so we can say that good selectivity towards the breast cancer cell lines for this, but not MCF7. For MCF7 is below 2. Below 2 is considered not selective. Selectivity is not good. Okay, and then okay, this is from here. 1A and 2A, they show acceptable inhibition towards this cell when exposed to cell line for 24 hours. So looking at the Lipinski rules, the values on there, the molecular docking values. So we have this 1A and 2A. So the binding energy is not that good. It's not that good. And also the inhibition constant is also not good. Okay, inhibition constant has to be low. And further investigation on the molecular docking work is still ongoing, right? So the 2D structures of the interactions of these compounds, 1A and 2A, will also be determined. Okay.
Professor, apologies for the interruption. There are 5 more minutes. Thank you.
Okay, all right. So I'm finishing already. Okay, so all these pyrazoline compounds we synthesize from a ring closing reactions of chalcone, right? And other ring also we have synthesized like pyrimidine and pyridine, yeah? And because we want to compare between all those heterocyclic ring compounds. Yeah? And series of starting materials of this chalcone. Okay, first of all, we have a lot of series of chalcone, and also we have published their cytotoxicity activity and also the molecular docking work. And type 1 pyrazoline compounds are still ongoing. Okay, instead of naphthalene, we also are working with anthracene, try to see how the additional conjugation of the benzene ring works. Okay, and also we are doing with coumarin-based compounds, right? And also imidazole. Some of the series are still waiting for cytotoxicity results. Okay, so I would like to thank my Malaysian government for the grants. Okay, without grants, we cannot do anything. Thanks to University Science Malaysia also for some grants, and thanks for the school for the facilities. No facilities is very hard for us to work. And also my collaborators, local and abroad, especially Indonesia, UGM, also UPI and UN University, Negeri Yogyakarta, UNY, right? And also in India and also in US. And my students, my colleague, and also my family, right? These are students from my, they are all graduated. No, only one waiting. This one waiting to submit his thesis, okay? Others already gone. All right, thank you. Thank you, Ms. Joanne.
Yes. Thank you very much for the great lecture, Professor. We truly appreciate it. Now, let us open the Q&A or discussion session. So for all the participants, please do ask the questions through turning on your mic through the chat box in Zoom meeting or the YouTube chat. Both in English or Indonesian are accepted. So please do ask away since we have Professor Melati Khairuddean here to answer the questions regarding the research on pyrazoline. Anyone, any questions?
Maybe you want to ask questions. Bolehkah buat research di Malaysia?
Boleh, boleh.
Boleh, kita kerjasama gitu.
Betul, betul, betul. Okay, Professor Melati, I have a question for you.
Yeah.
So I'm afraid that I missed your presentation before because could you please explain us about the how do you say why did you choose to use the thiamide in your structure, and also considering the Lipinski rule, just wondering how much the molecular weight of your bis pyrazoline.
Yeah. All right. Some of the compounds actually they are not like fulfilling all those data requested by the Lipinski rule. Some of it are very big molecular weight. But still, they are acting very good because some of our reference compounds, when we use, they also shows very high molecular weight, and still we are using it, right? Even log P should be less than 5, but sometimes it goes up until 6. Even tamoxifen also gives about 6. So we are there because we are now into this Swiss ADME, trying to understand using this, because there's a lot of softwares, because I'm working with a person in pharmacy, our collaborator in pharmacy, and they are helping me to understand, okay, so that we can know that, you need something that is polar here, not polar here, by understanding that, then only we can try to modify our compounds. Okay, so I can understand because I also question like this will be very high, so that is why we try to design our compounds that that has very small, that is small. When you have bis, then it will shoot, it will be very high. But then when you have bis, the binding will be better because it is bigger, larger, right? So the binding will be better. But then you will have like the molecular weight will be bigger because you are adding like double, you are doubling everything, right? So we are still trying to figure out those things. So halogen itself, when you go for bromine, bromine is like what? A lot. So then it will increase, right, the molecular weight. So those are the things that we need to really sit down and look at it and try to draw and figure out, how to go about what we want to do. Why are we adding here? Why? So if you can add only 1 chloro instead of 2, I mean, it gives this not very it gives more or less the same values. Just add 1. Why you want to add 2? Right? Because if you add 2, then you are adding like 35.5 into the system. And if you have bis, you are even adding more to it. So those are the things because I'm saying that the molecules are not all obey Lipinski rules, even though we say, okay, ideally you have to obey Lipinski rules or the rule of 5, so it cannot be more than 500, it cannot be this, cannot be that. So, but then, as you design, play around with it. And my students also, they are not patient students. All of a sudden, while I'm looking at it, they said, this is already synthesized, doctor. Oh dear. Already synthesized. Okay. They are not. They try to impress me by showing me after they did it. This is already done. Oh, I'm always trying to tell them, don't waste chemicals. Don't waste chemicals. So those are the things. And did I answer your question?
The last one, I think about the thiamide. Yeah, what is the role of thiamide in for the biological activities?
Thiamide. Okay. The substituent on the pyrazoline ring, is it?
Yes, yes.
Okay. We tried that and it shows maybe because of the sulfur, I don't know, because the sulfur can make the bonds. Sulfur pi bond. It can make the bond. Maybe it gives better. So we are still with that. We are trying to put trying to compare when you have sulfur and oxygen there. If you can see just now, we have C double bond S with NH2 and also the C double bond O with NH2. So we try to look at that, and we can see that when you have sulfur, you can make like interactions, better interactions lah, that will give better binding affinity. Even though it will be not significant, but then we are trying lah to understand more. We are trying to even put other substituents on the pyrazoline ring. And also when we have C double bond S and also NH2, we have in mind to build another ring there later on. So maybe can make another ring there.
Thank you, Professor Melati.
All right.
Thank you very much, Professor, for your answer. There are two other questions as we can chat box. Allow me to
How the mechanism.
question. I'm sorry, I cannot open my microphone because
It's okay. I can read here.
Okay.
How the mechanism of electron withdrawing group in pyrazoline structure and derivatives is better than electron donating. How about safety of pyrazoline due to the compound attack normal cell too? Well, I would say the mechanism part, we have our collaborator looking at the mechanism. In terms of mechanism of the chemistry, you cannot really relate with the what is happening in the cells, right? So we are working very close with our collaborator in terms of that, trying to understand the mechanism. I have one collaborator from Poison Center who are very good in doing this mechanism study. Okay, so I cannot really give you the answer of that, why the electron withdrawing group is better. If we have that, then we will include in our publication. Okay, so look for that. All right, so and then how about safety of pyrazoline due to the compound attack normal cell too? Well, in the drugs nowadays in the market, also they have a pyrazoline ring. They do have pyrazoline ring. It's just that the compound attack the normal cell. Well, that means it is not safe, because looking at that, to look at the selectivity means you are also looking at the drug safety also. You don't want your drugs to attack the normal cell. Because if it attacks a normal cell, then no point doing it. So you all the normal cell attack the cancer cell, good, attack normal cell, you die both. So you don't want that. So we are trying to look at it. Why pyrazoline? Maybe we are trying not only pyrazoline actually. We are trying with pyridine. We are also building the pyrimidine, trying to build the database for all those and see and try to compare between all those. But it needs a lot of time and students. Okay. On top of that is that we help. What is that? I'm sorry, I can't open. Okay, maybe you know Noviani. Oh, okay. Our research is about marine natural product. Okay, the 1st October we have about marine. Oh yeah, yeah. I know Dr. Noviani. Yeah. Why you choose natural product as the focus of your? Well, natural product is nature. I did natural product. I was with the group. I mean, I was doing the research work, but then isolation and everything. It was so frustrating. You need a very committed student to do it. Okay, because you don't need to isolate a known compound. You want to isolate a new compound. That is the challenge. Okay, so and then the students nowadays are not that how to say patient. They are not that patient. And then when you get compounds after column and column and column with all the methods and everything, so you still get very little product. Sometimes frustrating, not even enough for you to have all those characterization method being done on that. Okay, you have very little, and then you try to only can do NMR. Well, you cannot only do NMR. So that is why I slowly switch to the synthesis work. Because you have the idea already, and then this is what you should be getting. You don't really need, you just need NMR. The most you just need NMR just to confirm whether you get your compounds or not. But still, you are focusing on the scaffold of the natural products. Try to do that so that it becomes, you are still with it, but you are not doing it. Something like that. Okay, so for the student, please sampaikan salam kepada Dr. Noviani. Okay, Joanne.
Okay, thank you very much,
Right.
Professor Melati Khairuddean.
Okay.
So that is all for today's discussion session with our professor. Once again, many thank yous for the lecture and the new information you have shared with us today, Professor. So moving on to our second speaker, which is just as wonderful. Allow me to introduce Professor Diwan S. Rawat from the University of Delhi, India. Professor Rawat obtained his master's degree in chemistry from Kumaun University, Nainital in 1993 and secured first position in the university. He did his PhD in medicinal chemistry from Central Drug Research Institute. Once he worked at Indiana University and Purdue University, USA, and he was appointed as full professor in the year of 2010. Professor Rawat is a visiting professor at Japan Advanced Institute of Science and Technology, or JAIST. Professor Rawat has also been an associate editor of RSC, Advances and Nature Scientific Reports, and international editorial board member of GBMT. He has published over 157 research papers and has published a book, 4 book chapters, and has 9 patents to his credit. One of his molecules entered in clinical trials for the treatment of Parkinson's disease. He has been elected as the president of Chemical Sciences section of Indian Science Congress for the year 2019-2020, and he has received numerous awards and honors, like the Special Appreciation Award for Exemplary Services by University of Delhi 2021, among others. Professor Rawat has served the university in administrative positions like warden or provost of Jubilee Hall for nearly 16 years, and presently he is working as the dean of examination of University of Delhi and initiated the digitization of his examination process. And as the dean of examination, he created history by declaring all UG results more than 1 lakh students in record of 28 days time. Well, okay, the CV of our second lecturer is very extensive and remarkable, and we are very lucky to be here today and be able to learn from Professor Rawat. So without further ado, please welcome Professor Rawat. Good afternoon, Professor. How are you today? I'm sorry, probably you may unmute your mic.
Oh, yeah. Am I audible? Thank you very much for your introduction.
Yeah. Good afternoon, sir.
Yeah.
So for the next 40 minutes, the time is all yours.
Okay, thank you. Let me just share the slide. So can you see the slide?
Yes.
Okay. Let me thank Dr. Idham first for his kind invitation to me. I think a couple of years back, I intended to visit the university, but at the end, because of some family issues, I have to drop the program. Now today we are here we are meeting virtually, and this is what Corona has taught us actually. Most of our activities are nowadays online, so at least we can be in touch with our friends and make new friends in the academic world. Today I'm going to talk about some of the work we have been doing on medicinal chemistry. Before that, let me just tell you about the university. The university was established way back in 1922, actually. So we are celebrating 100 years of existence next year. And in the beginning, there were only 3 colleges in Delhi. That was St. Stephen's College, Hindu College, and Ramjas College. All these 3 colleges were affiliated with the University of Delhi because this was established at the time when Britishers were here. So in the British system of education, you have a university, and the university has a campus colleges. So that concept, these 3 colleges were affiliated with the University of Delhi. We started with a very modest beginning with the 3 faculties and 750 students. And today we have 16 faculties, 86 academic departments, 77 colleges, 5 other recognized institutes spread all over the city with 160,000 regular students and another 270,000 students take their education in a non-formal mode. And the administration of that huge system, and we have 10,000 faculty positions and about 25,000 non-teaching staff. And the administrative part of this huge system is looked after by the university, and examination branch look after the examination of all those students, and which I'm hitting right now. So at a time when we conduct the exams, our size of the examination is about 250,000 students at a time. Let me just show you some of the pictures of the university. This is the entrance gate of Delhi University. We have different gates actually. It is an open campus. And this is the administrative block of the university, what we call a Viceroy Regal Lodge, because this was the building was occupied by the Britishers when they ruled India. Till 1933, the Viceroy used to operate from this building. In 1933, the new building was initiated, and that's what we call is the Raishana Hill, which is now occupied by the President of India. So in 1933, this building was given to University of Delhi, and it has huge historical background, historical importance, because some of our freedom fighters were kept under this building, and many of the meetings and all those used to happen in this building. And this is the arts faculty. This is how it looks like, and this is the aerial view of the St. Stephen's College, what I talked to you a little bit earlier. With this note about the university, sometime if you have visit India, please make a plan to visit University of Delhi. We'll be very happy to host you. With this brief note, let me just talk about a little bit about drug discovery. When we talk about the drug discovery, historically, all the diseases and pains and all those kind of stuffs in the ancient days were used to be taken care by the plant cell. Till middle 19th century, all the diseases and human pains were cured by using the plants. The oldest written evidence of medicinal plant for the use of treatment of any kind of sickness of the human was documented on a Sumerian clay. The picture, what I have given over here, it is about 5,000 year old, and that was found in Nagpur, the city of one of the city in India that is called Nagpur. It comprises 12 recipes for the drug preparation, referring over 250 various plants and some of them alkaloids such as poppy, henbane, and bentonite. So this is the oldest written document which talks about the use of medicinal plants for the treatment of the disease. And in the Indian Vedas, we call it the Shustra Samhita, and this is the written language, so which I also can't read it because it is in Sanskrit. So what it says about 1000 to 500 BC, the Sushruta Samhita is one of the fundamental textbook of Ayurveda or Indian traditional medicine. The surviving text contains the prescription of 1120 illnesses, 700 medicinal plants, including safety, efficacy, doses, and benefits. So these are the old way of treatment. But when we talk about the synthetic medicinal chemistry or synthetic drug discovery, the oldest molecule that was discovered to use as a synthetic for the treatment of kind of a sickness that is called sedative hypotonia is a chloride in 1869. Then comes the antipyretic and analgesic, that is phenacetin and acetonolide, and they were the byproduct of the coal tar actually. So these were the second generation of synthetic molecules that came to the existence. But the most important one among them was aspirin. As we know, willow tree was used for ages in 300 BC for the treatment of the pain. But the synthetic molecule, which we call aspirin, was developed by Hoffman in 1897. So from the point, the natural product and the synthetic chemistry, how much the suffering and how much the advantage of that in terms of the disease cure took place. Let me just show you this slide. At 1900, the average age at birth in India was about 23. And Indonesia was little bit ahead. Now today, in 2000, the average age in India at birth is 65, and Indonesia stands over here, maybe 67, 68. So see, at 1923, in 100 years, we crossed 65 in India. The reason is chemistry played a pivotal role in this. Because the synthetic drug discovery program, the systematic investigation of the plants started from that point. So when we talk about drug discovery, this is the biggest nightmare, because when we talk about the drug discovery, it is very challenging, time consuming, and it takes a lot of efforts and money as well. So out of 10,000 compounds, if the medicinal chemist synthesize or isolate from the natural source, one compound comes to the clinical market as a drug. It takes 15 to 17 years in the development process, and it costs about $460 million US. So it's very, very costly. So this slide saves medicinal chemists whenever they deliver a talk. So at the end, nobody should ask the question, tell me if you have developed any drug or not, because this is the answer. It is very costly actually. Anyway, so let me just move what I wanted to talk today. We work on a concept that's called hybrid drug discovery. When we talk about the drug discovery, you know, when somebody falls sick and you go to the doctor, they will prescribe you more than one drug for the treatment of one single disease. The best example is the tuberculosis. Tuberculosis cannot be cured with one drug. You have to take more than one drug, 2 or 3 drugs at a time. So this is what it is called a combination therapy. And this combination therapy has met a success, reduced the mortality rate, cured many deadly diseases. But the problem of the combination therapy is manifold. One, when you take 2 compounds in a biological system, the metabolic stability and the metabolites cannot be same. It will be different, number 1. Number 2, there can be a chance of drug-drug interaction. Number three, this combination therapy has one major disadvantage, that it has helped in developing the resistance against existing drugs. So as a scientist, you can think like this. You have drug A and you have drug B. Okay, so just think about it. If you can hook up these two drugs by a covalent bond like this, when you do this, what happens? You have one molecule now. It's one molecule, but this one molecule has two drugs chemically bonded. Let me just show you the pictorially what I wanted to tell. So this is the combination. You take two tablets, or this is the tablet which contains two drugs. Now this one contains one drug, two drugs, and combined by a covalent bond. So it is a one molecule. That one molecule concept can be number one. So you have this red part and a green part. So these are two drugs. Hook up these two drugs by a linker. When you do that, so this is drug A, this is drug B, this is the covalent bond. So it means it's a one molecule. That one molecule, the what we call a linker, can be a stable one, cannot break down the biological system, or it gets break down the biological system, generates two different molecules. That's one. Number two, it can be a hybrid or it can be a merged. And hybrid concept may work against one target, or it may work against two targets. But if that works, so you will have an advantage. It's a like sword with double edge. You have a sword, you have an edge in both the sides. So that can help. And this concept of hybrid drug discovery was published first time in 1997, actually. This was the molecule. What was done from this part, this was one of the bioactive molecules, this was another bioactive. This part of the molecule was taken, and this part of this molecule was taken. And these were fused together and made this kind of compound. And people proved this compound was much better active than this one or this one. So during that time, actually, we started thinking as independent researcher. So we started working on it. But the best example is in the literature. It's in nature. So if you look at this plant, many of you must have seen. And this plant is a source of 70 alkaloids. Out of these 70 alkaloids, just forget about this purple line for a second. So if you forget this, then you have compound this and compound this. This compound is called pindolin, and this compound is called canelithide. Both of these alkaloids were isolated from this plant. Both of these compounds showed poor anti-cancer activity. But the same plant generated another big molecule when these are covalently joined like this. So that is called vincristine. And when you have N-methyl here, that is called vinblastine. And vincristine and vinblastine are blockbuster drugs used for the treatment of leukemia, and FDA approved this in 1963. So this knowledge was already there through nature. And medicinal chemistry did not utilize this for good 40-50 years. Now I will just tell you one more example. So, this is a chloroquine. All of us know that this is a treatment. This is used for the treatment for the malaria. And this is the molecule called artemisinin, which was isolated and developed by U22, who got a Nobel Prize for her discovery in 2015. And if you look at this molecule, so here you have a trioxane bridge. This molecule has a trioxane bridge here. And this part of the molecule was taken from a drug molecule, another drug molecule. When this was hooked up, this showed better anti-malarial activity than pharmaceutical industry in India, or Ranbaxy at that time, licensed this for the treatment of malaria. So we had some interest on malaria, and we wanted to work on a hybrid drug concept, what we did actually. So when we talk about anti-malarials, so anti-malarial class of compounds are divided in two different categories based on their mode of action. One is aminoquinone based. That aminoquinone, what it does actually, it inhibits the hemojoin formation. What happens when you get infected with malaria? So when the malaria parasite enters your bloodstream, it degrades your hemoglobin to heme and globin. Globin part goes down to amino acids. These amino acids are utilized by the parasite for its own survival because parasite cannot synthesize its amino acid. Now this heme portion, which comes from the hemoglobin, is highly toxic to the parasite. But parasite is very smart. It has a mechanism to detoxify. So what happens? This heme undergoes for polymerization, what is called hemojoin. So if you want to kill the parasite, you can do two things in that mechanism. One, stop the formation of amino acids. If there is no amino acid, parasite will die. Number one. Number two, if you can stop the hemojoin formation from heme, this heme is toxic for the parasite, will be killed by it. So this is what the chloroquine does it. So chloroquine stops the formation of hemojoin. That's number one. Number two, this is another class of compound called cycloguanine and pyrimethamine. Both of these compounds were used for the malaria treatment during 1960s. Nowadays, nobody uses it. What we thought, and interestingly, the target for the pyrimethamine and cycloguanine was DHFR enzyme. So what we thought, let's hook up these two pharmacophores together. What we did, we took quinoline part from here. So this is this, and pyrimethamine and cycloguanine, the part, the trizine or pyrimidine from here. This is what it is, and we have a linker. Simple. So one pharmacophore, two pharmacophore, and a linker. Let us see what happens. Let me summarize the result first. Initial result. What we did initially, so we took a trizine, then this kind of pharmacophore, and this. So if you look at, this is the activity of this compound. So this is the IC50 value of chloroquine. So if you look at the IC50 for our compound is much higher than chloroquine. So it means this series of compounds were non-active. Now, what was the fusion? Trizine, triazole, and aminoquinone. Now look at, this is the chloroquine. Height of this bar comes a little bit down. So it means the activity improves now. But when the trizine and aminoquinone were fused together, make a hybrid. Now look at the chloroquine IC50 value is this one. And our compound are much better active than it. So conceptually, we prove if you hybridize these kind of pharmacophores together, you can improve the antimalarial activity of those hybrids. When you work in a medicinal chemistry, this is the criticism sometimes the medicinal chemists get, because people mean the so-called synthetic organic chemist feel we don't do the hardcore chemistry because it's kind of a me too chemistry. You substitute one position to other position. So what we did, we substituted this nitrogen by CH. Rest of the things were kept same. So what happens? This becomes the pyrimidine hybrid. Let me just show you that synthesis. This was the dichloroquinone. This is a standard reaction. Then dichloropyrimidine was reacted. Two regioisomers formed, characterized by spectroscopic method, and we got the X-ray crystal structure. And these kind of compounds were made. Now, let me just show you the biological response, what happened. Now, if you look at this compound versus this compound, what is the difference here? The difference is the point of attachment of aminoquinone part to pyrimidine. Look at it here. It attaches at this carbon. In this case, it attaches with the carbon, which is flanked by 2 nitrogen of pyrimidine. Now look at the antimalarial activity. It is almost identical. So if you remember the previous slide, one was the major isomer, another was the minor isomer. So think about if the minor isomer shows better activity, the students fail because they have to do more hard work. So luckily we got the anti-malarial activity for both the isomers same. So we went ahead with the major isomer and functionalized it. This here is substituted by this heterocyclic amine, number one. Now the activity improves tenfold. Go again, morpholine, activity remains same. And ethyl piperazine and methyl piperazine, the activity is at the level of 7 nanomolar now. So once you have a 7 nanomolar activity, people will talk about its toxicity. Those compounds are non-toxic, number one. Number two, then people will talk, what about the in vivo efficacy of those compounds? Let me just show you. So these compounds, the chloroquine, 100 milligram per kg body weight for 3 days, there was zero cure, no cure. But our compound, 30 milligram per kg body weight, 3 days dose, 80%. We published this way back in 2012 in ACS Medicinal Chemistry. So once you have these kind of data, then you would like to modify what we call structural optimization or structure activity function studies. So we did all that. So this was the beginning compound, what we started with. From this, from 2.45 micromolar, we ended up with 7 nanomolar activity, and these were some kind of structural variation we did. We made about 700 synthetic compounds in that study. I will show you few of those studies. The first change what we did, we wanted to look at the effect of this linker. So these two pharmacophore, one is aminoculin, another one is the pyrimidine, were hooked up by a rigid linker, piperazine. And in another case, piperazine was kept there, but there was a flexibility of CS2 unit. Let me show you the biological response. What happens? This compound was our original compound of 7 nanomolar activity. When we put a rigid linker between these two pharmacophores, activity drops. But when the flexibility was kept, but piperazine is there towards pyrimidine, it regains the activity. It improves the solubility of the product. Not only that, the in vivo activity says 100%. With this, we did some kind of mechanistic study to show what is the mode of action of our compound. So we titrated our compound with heme. So this is the heme absorption maxima at 402. So when we titrate our compound with this heme, because if you stop the heme to hemojoin, you can improve the activity. What we show here, the activity, the interaction between our compound and the heme is very good. So by job plot, what we showed that the interaction of heme and our compound is 1:1. We did some molecular modeling studies to show that whether these compounds are binding in the binding pocket of DHFR or not. And this is the native DHFR ligand. And this blue compound here is the pyrimethamine. So if you see, it doesn't bind well in the binding pocket, but where our compound, this is our compound actually. So our compound binds well with the binding pocket of this DHFR enzyme. That may be the reason of better activity of our compounds, though experimentally we could not prove because we could not get a good collaborator who could do the DHFR activity on those compounds. Next, I will just show you one beautiful example, design-based example actually. This is a compound called amodiaquine. It was considered to be better anti-malarial than chloroquine. Was introduced in the clinical market in 1960s or so for the treatment of malaria, but it was withdrawn later on because it has hepatotoxicity. What happens when you take this molecule, it metabolizes in liver and it forms this kind of a metabolite, hydroquinone type. And this hydroquinone type or metabolite binds the liver cell and causes the liver toxicity. It's interesting to note paracetamol, what we take for cure of pain and the fever, has the same kind of toxicity. 37% of the paracetamol what we take, it metabolizes in the liver and forms this kind of metabolite, which is liver toxic. This is why paracetamol is not prescribed when a person has liver disease, because it damages the liver. But this knowledge, when the amodiaquine was developed, was missed by the chemist and the biologist. As a result, even though they had a very good molecule, it was withdrawn. If you remember this slide when I showed the duration and number of compounds, the money. So if you develop something and you withdraw it back, you can think the fate of that industry. You can stop the toxicity, remove the toxicity of this molecule. How? Either you switch off the position of these two. If you switch off the position, you will not get this. But whether the activity will remain or not, that's another $1 billion question. Another thing is, you can substitute this with a group which is of a similar size and similar nature, so that you stop formation of this metabolite. That is a fluorine. If you add a fluorine here, you will stop this metabolite formation. That was done. And prove non-toxic, number one. Number two, activity reductions. So what we did here, we took this portion of amodiaquine, only this portion, this fluoroamodiaquine, this N-diethylamine part was substituted by the pharmacophore what we had from our previous study, that is pyrimidine. We did that. What was the change? This is amodiaquine. The only change between amodiaquine and our molecule is this triangle part. This is OH2F and this one N-diethylamine. That's it. We characterize it. Look at the activity level. Chloroquine's IC50 is 220 nanomolar and our compound is 4.6 or 4.7 nanomolar. Less than 10 nanomolar compound from this range we have. The compound was 47 times much better than chloroquine. Now 100% cure, 33.53 milligram per kg body weight for 3 days. The metabolic study says the compound is much better stable than amodiaquine, which we expected. And we published this in ACS Medicinal Chemistry latest 2 years back. Another modification what we did now, this portion, fluorobenzene ring, was substituted by cyclohexane. That's the only difference between this molecule and this molecule. Now the activity is 2.2 nanomolar. Improve that activity. What about in vivo? The 60 milligram per kg body weight, but the treatment was for 7 days, but the mice survived up to 60 days, 100%. Not only that, they became healthier. The body weight of the mice increases. So complete paramecia clearance. This was the paper which we published in 2012. What we knew about chloroquine through literature at that time. In 1987, there was a paper published by Sanders in Nature, which says chloroquine can retard the Parkinson progression. This was the conclusion of the. Let me just show you this. This is what it says. Now, this is the chloroquine, and this is our molecule. Our molecule are much better than chloroquine in terms of anti-malarial activity. So we got interested to test our compounds, screen our compounds for Parkinson. You need some good collaborator for that. One fine morning, I received an email from Professor Kim from McLean Hospital, Boston, He said that they have a high throughput screening system developed, and they had screened 650 FDA approved drugs. And what were their results? Out of 960 FDA approved drugs were screened, and 3 hits were identified. One, amodiapan, chloroquine, and gliflozin. All these 3 has aminoquinone formula. So we got the opportunity to screen our compounds for Parkinson. We started collaborating with him in 2012. These are our molecules. Now, what is Parkinson? It is as we all know, it is a neurodegenerative disease, and estimated 10 million people worldwide have this disease. And the common symptoms are tremor, rigidity, slowness of movement, and difficulty with walking. The motor symptom of PD results from the death of neuron in the substantia nigra. This is the portion of a brain, this tiny portion of a brain. If there is a problem, you develop the Parkinson. Unfortunately, there is no drug for the treatment, for its treatment. Only L-DOPA is there, and that L-DOPA is manages. But once you have the disease in the advanced stage, you cannot have any effect of L-DOPA also. As you know, in our brain, we have 60 billion neurons. And these neurons have a beauty. They are not connected to each other. They are connected by a system called synapses. So synapses is something like this. So you have ion exchange over between this. So now if you talk about the synapses with 60 billion neurons, so you can multiply how many synapses are there. And that's the reason to develop anything or a disease like a brain disease is very, very tough. And there is one receptor that is called NER1 receptor. What this NER1 receptor is? Until now, till 2015, people thought NER1 receptor does not have a binding pocket. It's called orphan receptor. So if something does not have a binding pocket, you can't do anything. But Professor came, though the structure is not known yet, experimentally proved its binding pocket is occupied by a bulky amino acids. So there is very little space. But there is one mystery molecule which binds with this. When it binds, its flexibility improves. As a result, when NER1 gets activated, it helps to stop the death of dopamine neurons, which generates the dopamine. Not only that, it stops it cures the neurons which are dying because of Parkinson. That he published in PNAS. So that was the concept. So we started working on this. Initial studies showed, look at this. This is the 3-nitrogen, triazine, this is pyrimidine. These compounds are totally inactive. So initially we sent about 120 compounds in 2012 or 2012. And this was later funded by MJ Fox Foundation for this work. But these compounds are inactive. This were inactive. This were highly active. Now look at the EC50 value of chloroquine. It's a 50 micromolar, which is considered that it can retard the Parkinson growth. Now look at our compound. Our compound EC50 value is 0.5 nanomolar. 650 compounds were tested, out of which 15 hits were identified and 3 compounds were chosen for further development. What was that? Let me just show you whether this is activating the NER1 receptor or no. This is what the data is. So what we did the luciferase enzyme inhibition assay. In that, our molecule help to bind with the binding domain of nerve 1. The activation of nerve 1, the concentration. Chloroquine 100 micromolar. This is the activation. Our compounds concentration is 20 micromolar. So our compounds were 5 times better than chloroquine. And this is the whole cell assay, which shows you have a nerve 1 whole, and did the assay, we found the same thing, 5 times better. Immune suppression. Our compound has a very little immune suppression effect, while the chloroquine and the LPS has a highest immune suppression when you treat the Parkinson with that compound. And this is the neuroprotective effect of chloroquine and our compound. This is the cell with the vacuum, so you can see the green and red cells. So this is the compound that causes the Parkinson. So when it causes the Parkinson, you can see the red spots disappears, all green. So it means the disease. This is the LPS, another compound that causes the Parkinson. Now, when you treat this with chloroquine, the red spot starts appearing. But when you treat the same system with our compound, you see more red. So it means it goes to the better side. Similarly, with this compound also. So our compound has much better in vivo potential than chloroquine for the treatment of Parkinson's. This is the animal model studies, actually what they did. L-DOPA was given 50 milligram per kg body weight. Chloroquine 40 milligram per kg body weight. Our compound 5 milligram per kg. So 50, 40 and 5 milligram. The effect were better in case of our compound in comparison to chloroquine.
Excuse me, Prof. Pardon for the interruption. There are 5 minutes left.
Oh, yes. I will respond. Thank you. Now, let me just show you the brain structure of the mice which were treated with our compound, chloroquine, and the vacuum. This is the cerebral part of a brain. This is the healthy brain part. And this is the substantia nigra part, the healthy one, with these two features. When Parkinson's disease was caused, look at the structure of the brain now. You can compare these two. Now, this is treated with L-DOPA. Look at the difference. Little bit better. When treated with chloroquine, 40 milligram per kg body weight dose comes closer to vacuum. Look at the compound what we have developed. This is identical to the healthy animal. The dose level is 5 milligram per kg body. The body weight of the animal improves during the treatment. That shows the positive effect. These are the pharmacokinetic data of the compound what we developed, much better than chloroquine. With this is the major breakthrough. We have 20 compounds with IC50 value less than 15 nanomolar for malaria model, but 3 of our compounds have entered to clinical trial now. So this is the Norman Pharmaceutical located in Boston. We just a few months back signed the technology transfer agreement. These are 2 of our compounds. This is almost clear the preclinical trial, and this is the preclinical trial now. And we have a pipeline molecule of 15 another molecule which can be taken off if anything goes wrong with these 2 molecules. We work on green chemistry nanocatalysis that we started a bit late actually. Let me just tell you this Parkinson's disease. We have filed 3 patents, but till date we don't have any publication yet. So you can think to motivate the students to work up in an area where you may take 10 years or more to get 1 publication. It's very difficult. It's very challenging. So in between, we started side by side the catalysis work, and we published many papers on this green chemistry area, and some of the work has been highlighted by SynFact, appeared in the cover page of the journal. What I presented you today is because of the hard work of these many students who have worked with me, and most of them are well settled in their respective fields. Some of them are in the academics, most of them are in the industry, and some of them are doing postdoc in United States. And this is the research group. Last year, last year, January 2020, when I was Indian Science Congress sectional president, my whole group attended that because they had no choice because it was organized in a different city and I had to take care of it. The funding from all funding agencies in India and MJ Fox Foundation from the United States, highly appreciated. Thank you very much for your kind invitation and patience hearing. I'm very happy to answer any question if you have it. Thank you.
Thank you very much, Professor Rawat, for the excellent presentation. It is very, very detailed and interesting. So let us open the Q&A session or discussion session. For the participants, please do not hesitate to ask questions. You may turn on your mic, chat box, or the YouTube comment. Both English and Indonesian are accepted. Thank you very much, Professor. We learned plenty from your presentation today. So anyone who wants to ask questions regarding the presentation?
Okay, perhaps for me, Mbak Juan.
Yes, please, sir.
So thank you, Professor Rawat, for a very interesting presentation. So I have several questions for you. So the first one, yeah, is there any guideline to choose the linker in the hybrid molecule? And then the second one, what is the advantages and also disadvantages of the rigid and the non-rigid linker?
No, I couldn't get your second part of your question. Can you explain?
Okay, so you have explained before that you use the rigid linker and also the non-rigid linker, and you see the different activities from both. Yeah. Could you please explain about both advantages and also disadvantages of this linker?
Yes, actually, when we started working on this, because the flexibility of a molecule may affect the activity of the resulting hybrid. So we started with C2 linker, that is ethylene. Then we went up to C6 actually. Okay.
Yeah.
So from C2 to C6, the best active compounds were from C2 and C3. So when you increase the flexibility, the activity starts dropping it. How exactly that happens, why that happens, we have no answer for that. It's very difficult to correlate the biological data in terms of the scientific explanation. But yes, if you increase the more flexibility, the number of carbon atoms between two pharmacophores more, the activity drops. The optimum was 2 to 3.
Okay. So is there any guideline to choose a linker?
No, there is no guideline.
Okay. Thank you, Mbak Juan.
Thank you, Pak Idham. Are there any other participants? Oh, there's a question in the chat box from Ali Bahri Lubis. Would you like me to read the question or can you see it in the chat box, Professor?
What is the question is, could you explain me in general insight about the correlation between knowing mechanism of enzyme of heme deoxygenase and healing cancer disease? No, cancer disease actually we don't work on, but heme detoxification we also work on only that part because our molecules have a pi stacking. That pi stacking, when you have a heme heme structure, so it's a fat molecule. So your chloroquine comes on the top of it. So when it comes, so another heme molecule doesn't come. So the hemojoin formation, hemojoin is a polymer. So that stops. So once that stops, the free heme kills the parasite. But for cancer, how it works, I don't know.
clear enough, I guess. For the participant, Ali Barilubis, is Professor Rawat's answer clear enough?
Yeah, thank you so much. Actually, we have a executive council meeting of the university today, so which makes all the policies and everything for the university. So I'm the member of that committee. I just took off so to give this lecture. So after this, I will be going again to that meeting. Actually, thank you very much for your invitation and patience. Thank you.
Thank you. Namaste.
Okay, thank you very much. If there are no other questions for Professor Rawat, so that is all for today's discussion session with Professor Rawat. Once again, many thank you for the lecture and the new information you have shared with us today, Professor. And next, we have our third speaker from Sardar Patel University, India. Let me introduce Professor Hitendra M. Patel. Professor Patel is a professor specializing in organic chemistry. He received Doctor of Philosophy in Chemistry from Sardar Patel University in the year of 2008 and has been a professor since October 2016. His research area includes organic synthesis, sustainable chemistry, synthesis of various types of dyes and their intermediates, among others. Professor Patel has 30 publications and counting, as well as published several books. The highlights of his achievement includes elected as the member of Organic Division of Council in Royal Society of Chemistry UK, worked as an expert reviewer in many high-repute international journals, reviewer panel member in RSC Advances Journal UK since 2020, delivered many invited talks at the national and international level in conferences and universities, respectively. Also act as a review editor in Frontiers in Chemistry, Organic Section, Switzerland, editorial board members in Frontiers of Chemistry, Switzerland, and Archivoc USA, as well as worked as junior scientific assistant in Gujarat Pollution Control Board. So that's the CV of our third lecturer, which is very impressive. And it is an honor for all of us to be able to learn directly from Professor Patel. So please welcome Professor Patel.
Yeah. Thanks, John, for a wonderful introduction.
You're welcome. Good afternoon.
I need your permission to share my slide. Can you hear my slide?
Yes, sir.
Okay. So first of all, thank you very much. Thank you very much to the organizers from this Gadjah Mada University, Indonesia, and Aix-Marseille University, France, for this world-class professor series lecture program. So thank you very much for giving me opportunity to speak something with your scholar and the students. As you know, the Professor D.S. Rawat already explained wonderful thing based on the discovery of a drug molecule with highlighting the quinoline as well as the pyrimidine nucleus. And in earlier lecture also, the fantastic lecture explained by one of the speaker based on the indole moiety. So here I will explain the important concept, the sustainable approach for the ionic liquid mediated synthesis of the heterocyclic scaffold. So today's my talk will cover the general introductions of this talk and the sustainable approach for the ionic liquid, then the applications of the ionic liquid for the heterocyclic scaffolds, which includes the 2-hydroxyethyl ammonium acetate as the ionic liquid for the domino novenazole and the Michael addition type reactions, and the pyridine 2-carboxylic acid we used as a ionic liquid for the synthesis of pyrazole 3,4-B quinolinones. And third application I will discuss with you based on the piperidinium acetate for the designing of a CC and CN bond formation. And lastly, I will discuss the calculation of a green matrix for the multi-component reactions. And finally, the conclusion. Let me try to explain some of the introductory part based on the ionic liquid. When we talk about the ionic liquid, definitely the question came into our mind, why ionic liquid have their own importance rather than the volatile organic solvents. And as we know that the volatile organic solvent increasing air pollution for many chemical process, and volatile organic solvents cannot be easily separated from the desired reaction product, and it is difficult to recycle. And view of the awareness of the weakening that environment, researchers are directed on developing alternative environmental friendly solvent system to replace the traditional volatile organic solvents. And within this context, the interest of ionic liquid as a green solvent resides in their extremely low vapor pressure and the high thermal stability, which offers advantages such as the ease of control and product recovery and recycling ability. So friends, as we know that the important characteristic properties of the ionic liquid, with the simplest example, you can see here the salt of NaCl, where we found that the symmetrical anion and symmetrical cation. And on the other hand, if we talk about the ionic liquid, you can see here the symmetrical ion definitely we have. And of course, we can have the asymmetric cations. So that's the great combination of the symmetrical anion as well as the asymmetrical anions, which leads to the wonderful applications of the ionic liquid. So, so the ionic liquid is a generic term for the class of a material and consisting entirely of an ion and being liquid below 100 degrees Celsius temperature. And they are liquid at a room temperature, then we call they are the room temperature ionic liquid. So with these basic terms, I will explore that the some of the ionic liquid with having the variety of the cations as well as the anions. So as you can see that the cations we have listed here with the 5-membering system, 6-membering systems. So there is nothing but 1,3-dialkyl imidazolium, N-alkyl pyridinium, tetraalkyl ammonium, and N-alkyl pyrrolidinium. So these are the cation we have, and the anions we have to select it based on requiring that the hydrophobic nature as well as the hydrophilic nature. If we need to explore that the hydrophobic natures, then we have to use the hexafluorophosphate PF6 minus TFSI, and here the tetrafluoroborate, and here we have to take the trifluoromethane and sulfonate, trifluoromethane sulfonate, and nitrates if we have to go for the hydrophobic properties, then chlorides and bromides like that. So these are the general characteristic properties of the cation and anions we have to select. So the ionic liquids can have their own advantage, that is only because of they are tunable in the wide range, as well as they are having the high stability and instinct conductivity we have, and no volatility with this ionic liquid. So that's why I will try to explore that the types of ionic liquid. So it may be a task-specific ionic liquid, switchable polarity solvents, the chiral ionic liquids, polarizable ionic liquids, the metal salt ionic liquid, bioionic liquids, supported ionic liquids, and the protic ionic liquids. And another great advantage we have with the deep eutectic solvent, it is a little bit different rather than the ionic liquids and the amphiphilic ionic liquid. So here I covered the basic concept based on the ionic liquid. I inserted the many slides before that my actual work. That is only because I have seen that many graduate and undergraduate people also joining with this meeting, so they can understand the background of this ionic liquid with the basic concept. So that's why I inserted these important things. As we know that the broader applications of the ionic liquid we have in the engineering side and the electrochemistry base and the physical chemistry, as well as the solvent and catalysis and the analytics and biological sides, we can use these broader applications based on that the important characteristic properties. So that's the things we have with the biological applications of the ionic liquid. So let me try to explain the advantage of the ionic liquid. So here the easy separations we have to have with the ionic liquid whenever we have to utilize in organic synthesis. Then the very low vapor pressure we have, and non-flammable substance we have, and high thermal stability we have with the ionic liquid, even though we can have the high mechanical and the electrochemical stability we have. And that's why the ionic liquid have their own advantage. And the most important advantage of the ionic liquid, they are showing the low toxicity as well as non-volatility. So non-volatility, and that's the great advantage of the ionic liquid. So as we know that the little bit the introductory part of the ionic liquid, we have to how we can focus this ionic liquid for the many organic transformations with utilizing different kind of ionic liquid. So when we have to utilize this ionic liquid for the synthesis of an organic molecule, then definitely the many people, they use the word as the green chemistry and sustainable chemistry. So then definitely we have to keep this 12 concept in our mind for the green chemistry. One, there is a waste prevention. It must be required whenever we design a new work, and atom economy, less hazardous chemical process, and designing a safer chemicals, safer solvent, as well as the auxiliaries and energy efficiency. It must be required with whenever we have to design these work, and renewable feedstock, as well as the reduced derivatives, catalysis, and the designing of the degradations, and the real-time analysis for the pollution prevention, and the safety chemistry for the accidental prevention. We know this 12 principle concept for the green chemistry, but it's very, very difficult to implement on a design organic synthesis, specifically for the novel organic molecule. We design it and we proceed for the reactions. Then we keep certain things must be there with our reaction protocol. So then and then only we can fulfill this the sustainable approach for the reactions. Let me try to explain a little bit based on the solvent selections. As we know that the preferred solvent. So friends, here you can see that the water, acetone, ethanol, propanol, and the ethyl acetate, methanol. So these are the green solvent I'm talking about. We can utilize any of the reactions. There is no problem at all, and based on the selection of a solvent. And when we compare this preferred organic solvent, these are the usable organic solvent, not exactly the suitable to the sustainable chemistry, but we can utilize this solvent if it is required. If it is necessary, then we go with the cyclohexane, heptane, and the toluene, as well as the methyl cyclohexane, isooctane, acetonitrile, and xylene, and acetic acid, and ethylene oxide. So we definitely use this solvent. If we are not having any other option to use this one, so we can have a problem for the solubility and for the reaction efficacy. We may use this if it is required. But this is the danger zone which is shown here. If we have to use this solvent, then according to the guideline of a green chemistry, we cannot utilize this solvent like pentane, hexane, and diethyl ether, chloroform, dioxane, and benzene. So we cannot utilize this the solvent for the reactions. So that's the solvent selection concept we have to keep in our mind. Based on this green chemistry, we collected this the reference. Now, let me try to explain the solvent replaceable table. As I already explained, undesirable solvent we have that can be definitely altered with the heptane and heptane here rather than this hexane. We can use this alternate. We have these alternatives of the different kind of solvent for the reactions. So acetonitrile, triethylamine, the ethyl acetate, and the toluene. We may use this as an alternative instead of this kind of different solvent system. Now the comparison. So when we have to talk about the comparison of organic solvent with the ionic liquid, so you can see that the property here we have the number of solvent. So when we talk about the organic chemistry, we have whatever the opportunity we have to have for the organic solvent. So greater opportunity we have with the number of solvent selection for the ionic liquid rather than this organic solvent. And applicability is concerned for the organic solvent. It's a single function we have, but in the case of ionic liquid, we can have a dual role of this ionic liquid. We may utilize it in the reactions. And the catechetical applicability is concerned, we have a very rare, and similarly with the chirality also. But when we talk about the ionic liquid, definitely it is common one, and it is tunable. It is tunable, that applicability we have. Now, another thing, the vapor pressure is another important property for this ionic liquid. When we compare with this one, here we can, it obeys the Clausius, the Clapeyron equation, as well as here the negligible vapor pressure we have with under normal condition. And flammability is concerned, we have usually non-flammable of this ionic liquid other than the organic solvent, and that's a great advantage of the ionic liquid. So when we talk about the solvation properties for this organic solvent as well as ionic liquid, so here it is weakly solvating the property we have, whereas here we can have a strongly solvating properties. And when we talk about the polarity, then definitely we can have the polarity concept is a questionable one for this ionic liquid here, and as compared to the conventional polarity concept applied for the organic solvent. So that's the comparison of the organic solvent with the ionic liquid. Now, we focused mainly our research work based on the multi-component reactions with utilizing the ionic liquid as well as the deep solvent also. Today, I will discuss basically the background on this ionic liquid for the many kinds of organic formation, organic transformation. So the multi-component reactions is a powerful and convergent tool that employs the fusion of a two or more substrate, and it produces the product incorporated of almost all the atom present in the substrate. So the importance of the multi-component reactions in the organic synthesis, as you can see here, the greater advantage of the multi-component reactions is we have to have a high atomic economy as well as the reductions in the number of steps we do for the design work, as well as the access to the complex molecule convergences, as well as the easy of the aggregations and the short reaction span, which utilize our goal of the organic molecule, specifically for the synthesis. So that's why we can have the importance of the multi-component reactions. By keeping this in our mind, I will focus the nitrogen containing heterocycles, how we can utilize it, what kind of the catalyst we use it for the multi-component reactions when we have to go for designing of the nitrogen containing heterocycles by keeping sustainable approach in our mind. So friends, here also you can see that the huge the option we have as ionic liquid, the organocatalyst, and the group assisted purification chemistry, as well as the microwave, the option, and the nanoparticle, ultrasound, additive freeze, and the deep eutectic mixture. So we have a huge scope for this multi-component reaction with using different kind of that parameter. But I will focus mainly today with the ionic liquid based the catalyst for the multi-component reactions. Now, ionic liquid assisted multi-component reaction. Just I will share with you the history of that the ionic liquid. The many people they utilize for this kind of molecule. As you can see that the Rajani as well as the Mats who synthesized the bis coumarin molecule with taking 2-hydroxy ethyl ammonium acetate at 40 degrees centigrade temperature, and at 3 hours they converts into the bis coumarin. And another reactions, if I'm talking about here, then you can see here also you can see that pyrazoline derivatives we have. And another example, if I'm talking about the 2-amino-thiopyridine derivative can be synthesized from these 3 important components as melononitrile aldehyde as well as thiophenol. That work is reported by the Sobani Mats. At room temperature, they are able to carry out these reactions within the 5 minutes. So within the shorter period of time, they synthesize this pyridine nucleus. Here the Cho and the colleagues, they synthesize the bis pyrazole 5 all kinds of molecule with utilizing the same kind of that ionic liquid. And under solvent-free conditions, they reported these product formations at 90 degrees centigrade temperature. Now let me try to show you that the another example, the Sardar et al., who also synthesized this molecule in terms of the benzopyran with having 43 to 87% of the yield, while the pyranochromine is synthesized up to 98% of the yield with using this 2-hydroxy ethyl ammonium acetate in format in presence of by utilizing the room temperature reactions. Now here again, their work which is reported with the new formations, that is benzo 7,8, the chromino 2,3D pyrimidine. So that's the history based on this the selections of ionic liquid for making a different kind of organic transformation. So by utilizing the 3 different, I will show you today the 3 different kind of ionic liquid for the formation of a heterocyclic molecules. Let us see here the applications of ionic liquid for the domino nominal and Michael addition type reactions. So friends, here we have taken the amino derivatives of the pyrazole that can be interacted with this carboxyaldehyde, thiophenol based carboxyaldehyde, and 1,3-diene we have taken in a 1/4, and we carried out these reactions by using 2-hydroxyethyl ammonium acetate as the important ionic liquid, and we transform into this product. So you can see that the pyrazole based pyrimidine nucleus we synthesize in the systematic manner, and we reported, I think, here about 15 examples we reported for this kind of transformation, and we reported this work in the bioorganic chemistry in 2008, 2019. So let us see here for these reactions. I will discuss with you the optimizations reaction conditions for this one compound. So friends, here we have used many kinds of ionic liquid. We used many kinds of ionic liquid. There is nothing but tris hydroxyethyl ammonium acetate and bis 2-hydroxyethyl ammonium acetate, then lactate, formate, acetate. We utilize this one, and we found the best ionic liquid is the 2-hydroxyethyl ammonium acetate. We use for this organic transformation pyrazole-based pyrimidine nucleus. And we use 95 degrees centigrade temperature. Within the 120 minutes, we have 84% of the yield. So based on this optimization reactions, we design the 16 different samples. So as you know, the different hydroxyl alkyl ammonium ionic liquid, we use it for this study. So as you know that the format 2-hydroxyethyl ammonium format and 2-hydroxyethyl ammonium acetate, lactate, and ethyl acetoacetate form of this ionic liquid we prepare in our laboratory. And we found the best property of this ionic liquid we found. So that's why we utilize this ionic liquid for the whole reactions for 15 entries. Not only that, we prepare this ionic liquid and we check that the recyclability according to the sustainable approach we maintain over here. By taking this ionic liquid, we have a great recyclability of this 2-hydroxyethyl ammonium acetate as ionic liquid for our design reaction. So you will find that the fresh run we have to have 84% of the yield with 120 degrees and 120 minutes. And within 120 minutes, you can see that with the first run, we have 82%, and then with the second run we have 81% of the yield by increasing that time period. So you know that the productivity that can be decreased fall in this way by increasing that time. So that's the recyclability of the 2-hydroxyethyl ammonium acetate of ionic liquid for our design reactions. Now let me try to explain that the functionalized pyrrole-based dihydropyridine derivatives with the 15 entries by using this ionic catalyst. So I will try to show you that the mechanism. So this is the 2-hydroxyethyl ammonium acetate. We utilize it here. So as a catalyst, let us see here the aldehyde molecule that can be interacted with this the acetate ion, which will the 2-hydroxyethyl ammonium ionic ammonium acetate. We use the catalyst. So there is nothing but first of all the protonations is take place with this aldehyde, and we can have the protonated carbonyl carbon with the aldehyde that is happened. And then you know that the protonations, how it is happened, and then after the abstraction of the proton is take place over here from this active methylene, and then it makes a nucleophile species. When it makes a nucleophile species, that can be interacted with the carbon of this OH. Okay, and a nucleophilic attack is take place, as well as the CC bond formation is take place over here. You can see the CC bond formation, and after getting that CC bond formations, again the protonations is take place. When the protonation is take place over here, then after we have to go for the eliminations of a water. So removal of a water is take place, and then after we can have this the olefinic part formation here. So then later on, again, we have to interact with this, the third our component as a aminopyrazole-based molecule that can be interacted with this part. But before that, we have to go for the protonations from this ionic liquid. The protonations is happen here. After getting the protonations, we have to interact with this one. As far as the nucleophilic the species is concerned, it can interact with this one, and we can have the CC bond formation. After getting CC bond formations, that is happen via nucleophilic attack, we have the CC bond formation. The resonating structures which is shown over here in the keto-enol tautomerizations which is happened, and this step is very, very important to us for getting this reactivity to make the cyclization. After getting these important steps, we can have the cyclization that is feasible because the nucleophile species we already have with this NH2 that act as a nucleophile on the carbonyl carbon. As we know that here we can have the greater electron deficiency we have, so that's why definitely we can have the cyclization. After getting the cyclizations, we'll try to eliminate the again there is nothing but OH formation is there. Then the further the cyclizations is after cyclizations we have to have a protonation. After the protonations, it releases electron to eliminate the water molecule in systematic manner, and we can have our target molecule with the 15 entry. So then you can understand after removal of the water from the product reaction mixtures, we can recycle this molecule, this ionic liquid as a 2-hydroxyethyl ammonium acetate that we utilize further for the second run or maybe the third run. Now, let me try to explain that the designing of our work with using 90 degrees centigrade temperature and 120 minutes. Our target is to prepare this kind of molecule, but we didn't found this, but we have these important molecules. So the pyrazole-based pyrimidine dihydropyrimidine derivatives, we have it. So as you know that is based on the reactivity of the amines with the active methylene. So you know that how it in a different way it interact with this one. We check these things. We didn't found this kind of formation. Instead of this, we can have this 1,4 the interaction. I mean the carboxylaldehyde functionality with the fourth position of the NH we have rather than this. Right, so that's the ionic liquid which triggers selective one-pot reactions for this pyrazole dihydropyridine derivatives. Now, as you know that there is nothing but the 2 C bond formations, as we have seen that the mechanism 2 C bond formation and 1 C bond formation is take place, and we calculated with the all 15 molecules, we calculated that the green chemistry matrix we calculated, and we found out the E factor. When we talk about the sustainability, then E factor is very, very important part with having the greater amount of yield without wasting a byproduct. So that's the things we kept in our mind, and most of this you can see that the substrate scope how we have with a greater amount of percentage yield 79, 84, 87, except 3 molecules. You know that the 62, 60, and here 60. So these are the 3 compounds we have the less percentage of yield. Otherwise, the remaining that the 12 molecules have shown the very good percentage of yield. So now let me try to explain you. I will not explain that the proton and C13 and mass, all these things, but I will show that the crystal structure of our synthesized molecule. We develop the single crystals in our laboratory. You can see that the wonderful golden with the solvent, we can have a golden color of this the crystals they are developing, and we send for the analysis, and we have to have this R-type diagram of this molecule, right? And you can see that the systematic packing arrangement we have for this molecule. So we have to have a CCDC number. We deposited this structure, this crystal, and here also you can see that the CCDC number we quoted over here. This is another crystal we developed for the another compound, and you can see that the wonderful the packing arrangement of our synthesized molecule. So this axistic analysis of our developed crystal is enough for the identity of a molecule, how we synthesize. Now let me try to explain the other things. This is another example of a single crystal. You can see that just like a rectangle form, we have this R-type diagram. This is another the packing arrangement for this molecule. Okay. And here also you can see the wonderful the other the rectangle size. You can see the little bit having the solvent system. So that's why whenever we capturing that image, you can see that it looks like a golden color. And here you can see the R-type diagram as well as the similar the packing arrangement. You will find all this data from the BOC journal with elsewhere publishing house. Now let me try to focus some of the important activity based on this anti-microbial activity. MIC is reported over here with the help of collaborations. You can see here gram-negative as well as the gram-positive bacteria. With this the strain, we have to check with the standard drug as ampicillin, as well as the chloramphenicol, as well as ciprofloxacin and norfloxacin. We have to check this molecule, and we found out the few compound 4C, 4D, and 4I, which shows the good activity as compared to the ampicillin. So that is found out here. And as far as the fungal activity is concerned, we have to check with this 3 important strain. With the collaborations, we got this data, and we didn't found a good activity in terms of the antifungal for this our synthesized molecule. But of course, when we talk about the antibacterial activity, we have some good activity based, as compared to the ampicillin that is found out. Now, we check this anti-proliferating activity from the Spain, Professor Joseph Padron, who helped us for getting this data. So there is nothing but the GI50 value that is reported over here. So hello. So here we have the 6 different cell line. With these 6 different cell line, we check the standard drug as a cisplatin. When we have to compare with the cisplatin, only our 2 compound which is found out with the great potential, and that's why we reported this data as compared to the cisplatin. And let me try to explain this the structural activity relationship of this molecules. So there is nothing but this is the functionality we have. Whatever the substituents we have, it shows the good the antifungal activity. Here also, due to the presence of this functionality, we have a good function, the antifungal activity, and we can have a good bacterial activity if we have the substituents in this manner with these respective positions. And here the fungal activity we have good with having the CS3 substituents on the R1 and R2. And here you can see the bacterial activity is there with these important substituents respectively with this aldehyde molecule. So here this molecule, this substituents with these positions which shows the bacterial activity. And you can see here, these are the brief, the structural activity relationship which is shown over here with this our molecule. I will not discuss the whole the things, but you can see that anti-proliferative activity we have good with this the bromo containing this the subthiopine base 2-bromothiopine based aldehyde, which shows the good anti-proliferative activity also. And of course with this one also we have it increased anti-proliferative activity against the cervix and breast as well as the colon solid tumor cell line with the 4F compound. So that's the things we have based on the this the antifungal activity. Now let us see here the another synthesis. We synthesize the another molecules based on this pyridine 2 carboxylic acid we utilize as an ionic liquid. So when we talk about this molecule, you can see here how the pyridine 2 carboxylic acid we use as a catalyst with taking 10 mole percentage. With using ethanol, we use the 60 degrees centigrade temperature up to 2 to 10 minutes. We synthesize this target molecule. So friends, this is the aldehyde and this is the amino base. So it is a little bit different with using this different the catalyst. And with taking 2,1,3-diones, we have to have this important the similar kind of molecule with using different approach we synthesize. And the great advantage of our work is to source the rapid, the reactions is very rapid, as well as the reusable catalyst we utilize as this one. And there is no toxic waste we have. Even there is no metal involvement for the synthesis. No column chromatography is required, even though we can have a great amount of yield. And it's of course it's an eco-friendly route. We use it for this transformation. So let us see here the diverse route as according to the earlier reported works. You can see here this kind of with using the microwave irradiations, they use ethanol and base at a higher temperature with 20 to 30 minutes. They reported this kind of molecule. And here with using the pyridine 2 carboxylic acid ethanol as a solvent system at 60 degrees centigrade temperature, they synthesize this molecule, the pyrazolo 3,4-B phenolenones they prepared. Now, let me try to explain that the optimization reactions we carried out for this transformation, there is nothing but here you can see, I will not discuss, we tried with the other catalysts also, acetic acid and FeCl3 and betaine oxalic acid, even though we use the deep eutectic solvent also, but we found that great efficacy of our catalyst, that is pyridine 2 carboxylic acid, with using 20 mole and 10 mole. So we found out the great potency of our catalyst with using the 10 mole percentage we use and ethanol at 60 degrees centigrade temperature. Within 5 minutes we can have 100% of the yield. We try to increase the more concentration of the catalyst also, but even though there is we can have the similar kinds of the product we have. So based on this optimization reaction conditions for this our target molecule, you can see here the substrate scope. We have the variety of the substituents, not only that the hydrogen with the R1 and R2 and R3, but we use the methyl as well as the chloro as well as the methoxy group. Even though we had a combinations with the electron withdrawing group and electron releasing group also, and moderate electron releasing group also, we have to have a greater amount of yield that we synthesize. Now let us see here, the mechanism is same. I will not discuss this mechanism with you. As far as the recyclability is concerned, we have to have a 98% with the first run. With the second run, we have 95. Third run, we can have the 86. And with the fourth run, we have 80% of the yield. So that's the things we have. Now let me try. We will try to show that a comparison with our work with the earlier reported work. You can see here these are the reference we quoted with using this different reaction conditions, and here we can have the within the 3 minutes we have to have a 96% of the yield, specifically for the for this 4J compound, not with the 5J compound. So this is the target molecule we synthesize rather than this one. And the earlier people, they also reported somewhere with this reaction conditions, they reported this compound is also produced. But in our case, we didn't found this kind of product formation. So this is our work we shown here, and based on that, we synthesize this the 12 entry of this molecule. Now let me try to explain the another reaction concept. So whatever the earlier reactions I discussed, that we reported with the RSE advances. And here also this work also we reported with the RSE advances based on the piperidinium acetate we use as a catalyst for the CC bond as well as the CN bond formation. So as we know that for the synthesis of a heterocyclic compound by maintaining a sustainability, it's a very, very difficult task for getting the final product with a good amount of yield and with minimizing the waste product. So here we targeted that the formation of a 2 pyridone molecules. Why? Based on the existing drug molecules which is available into the market, based on these the 3 important drug molecule as a history, you know that it is useful for the treatment of idiopathic pulmonary fibrosis. This is the molecule have the important skeleton as a pyridone. There is nothing but the pyrphenidone name. This is the trade name. According to the trade name, you can have a different kind of name. So now milrinone, it is also useful for the treatment of a cardiotonic agent for the treatment of a heart failure. It is used, as you know, that the pyridone skeleton. Here also you can see that the pyridone skeleton with having different functionality with that respective positions. Perampanel, useful for the treatment of a Parkinson disease. So looking to this great the applicability in a medicinal chemistry, we thought that to design this kind of work. And let us see here the scheme. Friends, the little bit the interesting part I will discuss with you right now. That is only because of we found out the simplest chemistry from these reactions. So friends, we know that we have to take aldehyde and we treat with this melanonitrile. Suppose if we have to treat with the melanonitrile, then piperidinium acetate, we use it and we can have this N-substituted cyanoacetamide. Right. First, we prepare this N-substituted cyanoacetamide, and then we have to go for the further cyclizations, right? With using for the further cyclization, with using that the naphthyl phenyl ring system and the substituted phenyl ring system, we use it for the cyclization, and we found out the pyridone derivatives. So I think 12 entries we have with these pyridone derivatives, with having the 1.2-step reactions that we carried out. No column chromatography is required for the separation, and the milder reaction conditions we use it, and reusable catalyst. We shown that we reported that the pyridinium acetate is reusable, and we can have a good percentage of yield even up to a 4th run. So that's the future of these reactions, and we found out that whatever the phenyl ring system, substitute phenyl ring system, biphenyl ring system, even though we use a naphthyl ring system, then we can carry out this the pyrone derivatives easily. That is only because of the low steric hindrance is occurs. But on the other hand, instead of this one naphthyl ring system, if we have to use the enthryl ring system, then we found that due to the high steric hindrance, we didn't go for the we didn't receive the cyclized compound, but we received that N-substituted 2-cyanoacrylamide is obtained. So that's the important characteristic properties we observe in the simplest chemistry we highlight over here based on our result achievement. So you will find this work from the RSE Advances journal. We published in RSE Advances in 2020. So friends, let me try to explore these reactions as I already explained that in presence of piperidinium acetate at room temperature with using ethanol, within the 5 minutes we can have this important the olefinic formation. And then later on we have to interact with the piperidinium acetate by refluxing with the ethanol. We have this our target molecule. So I hope you can understand the formations. The steric hindrance cannot be happen, then definitely we can have the cyclization. But if you can have a steric hindrance in terms of the aldehyde, so then we cannot have the cyclization, but we can have the other product. Now let me try to show that the substrate scope. We have this kind of phenyl ring system, then there is no problem. Wonderful, we can have the product as well as the easy cyclizations happen within the 1 hour. But when we have to increase the bicyclic ring system, the biphenyl ring system, we use it as aldehyde. So then we can have a problem and it increase the time, and definitely it will decrease the percentage yield also because of the reactivity. That's the simplest chemistry we identified over here and reported. So if we have to 1 naphthyl, then also you can see that it requires the 1.5 hours and it degrades that the percentage of the yield. And another way, when we talk about the steric hindrance that is caused by this ring system, tricyclic ring system with the aldehyde, when we try to go for the cyclizations with this molecule, we cannot have the cyclization. But here we found out that this kind of molecule, that is 4 compound we observe and we characterize and we reported in the journal. That is Z and E isomer that we found out here. And you can see here that can be calculated from the LC chromatogram with the Z isomer retention time and the percentage area, and based on that we identify this is Z isomer, this is E isomer, specifically for this 4 component. So that's the things we have. And as far as the recyclability is concerned, we run up to 4 for having 61% of the yield. So within the 65 minutes we can have. So you know that with we have to have a degradation of the product with if we increase the runs for the synthesis. So hope you can understand this. And this is wonderful things we have in terms of the reactions. So as you know that we can have the melanonitrile that can be interacted with the piperidinium acetate. It makes the nucleophilic characters. When we make the nucleophilic characters, it can be interacted with this carboxylaldehyde. So it can be interacted with the carbon with carbon, and we can have a CC bond formation. After getting CC bond formation, friends, here we can interact with the further ionic liquid. So there is nothing but the protonation is also take place, and it eliminates the water molecule. After getting the eliminations of the water molecule, here it makes a double bond here. You find that this double bond. After getting this double bond, we have to interact with these our third molecules. In presence of piperidinium acetate, it abstracts the proton and it makes as a nucleophile, and the nucleophilic attack is take place on this carbon atom. After getting this one, we can have this part. So now here you can understand the hydrogen is added from this piperidinium acetate, and then after later on you can see it makes the this kind of. So when we talk about this cyclization, here we can have the cyano group. Right, so in a systematic arrangement, we did it, and the cyclizations, how it is happened in presence of piperidinium acetate, that you can understand. It makes a CN bond formation. There is a C double bond NH, and then you can see here it makes the double bond here, and it eliminates the freedom of NH2 here, and then finally we can have this kind of the final product. But when we talk about this static hindrance, if we found out, then up to this one, we can have the systematic things. But when we interact with this one, we cannot choose go with this side. But here it is happened. And in this pathway, we have to synthesize this and substitute to cyanoacrylamide. So this product is occurs when we have to have a bulky group if we use as the aldehyde. So then it reverses. So this route, which is shown over here very slowly to form this molecule. So this is the mechanism we have for this two important product. So let me try to explain. Yeah, how much time I have?
5 more minutes, sir.
5 minutes. Okay.
Thank you.
Thank you very much. Let me try to explain you that the green calculation of a green matrix for the multi-component reactions. So that is very, very important when we have to design any other organic synthesis. We'll try to see that the what about the green matrix for our design work. So let me try to explain. When we talk about the sustainability of our reactions, then definitely we have to take care about the, we have to identify that and measure that the effective mass yield, as well as the atom economy and the atom efficiency and reactive mass efficiency and the optimum efficiency, process mass intensity and mass intensity, as well as the mass productivity and E factor. This is very, very important. If we have a less E factor, then definitely we have our work is more strengthened one if we can have a low E factor. With using solvent as well as the water intensity, also we can carry out this E factor value. So suppose we design a work, then before that we use the reagent, we have to have a concentrate. These are so for example, if we have different kind of 3 important molecules, and we calculate that how much the weight we use for the reactions, that we have to notate over here based on the molecular weight. We have taken this molecular weight. As we know, it's a very simple thing. Similarly, whatever the reagent we use it, that we have to weight it properly before the reactions, and then finally also you can collect the product and you weight it, and then you have to think about the calculation based on the solvent you use it. For example, water in reaction media 3 ml 2.92 gram. You have to calculate how much that the weight of solvent that we use it, whether it is solvent or what as a water or as a ethanol or any other things that we have to keep in our mind. And based on that, we have to weigh this the molecules, whatever the final product we have, that we have a record, and then we have to utilize it for the calculation of a green matrix for our the final product. So let me try to explain you here. So there is nothing but atom economy can be found out with using molecular weight of the product upon total molecular weight of the reactant. into 100, we got the atom economy. That is very, very essential for the sustainable reactions. So if effective mass yield, that is also carried out by the mass product, a mass of the product upon the mass of the non as well as the winning reagent into 100, we can have the effective mass yield. Similarly, the atom efficiency that we have to carry out in presence of whatever the atom economy we have, that can be multiplied with this percentage yield, and we can have the atom efficiency. So similarly here, in the case of optimal efficiency, we use the RME upon RME upon the atom economy. I mean the reactive mass efficiency upon the atom economy into 100, we can have the optimal efficiency. Now, the another things we can have the reaction mass I cannot move. Yeah. So reaction mass efficiency is also calculated based on the mass of isolated product upon the total mass of reactant into 100. We can have the reactive mass efficiency. When we talk about the process mass intensity, that can be calculated with using the total mass of the input material into the whole process, including the solvent used for during the workup procedure, that we have to also count it upon the mass of the product. We can have this PMI. Now, then mass intensity and the mass productivity as well as the E factor that we have to calculate it. So when we talk about the mass intensity, total mass of the used in the whole process or the process steps, ignoring that the water contained upon the mass of product, we can have this mass intensity. Mass productivity can be calculated in this way. And as far as the E factor is concerned, without catalyst and with the catalyst, we can calculate in this manner mass of the raw material minus mass of the product upon mass of the product, which will give you without the catalyst, that E factor can be calculated in this manner. And with the catalyst also, we can calculate this with using the mass of the raw material minus mass of the product upon mass of the product. So solvent as well as the water intensity is also calculated with using total mass of the solvent, ignoring the water into the whole process upon the mass of the product. So solvent and water intensity is calculated based on the total mass of water useful for the whole process upon mass of the product. Whereas we talk about this, the water intensity can be calculated with using total mass of the water used the whole process upon the mass product. So friends, there is nothing but this is the calculation of a green matrix for our design work, that is found out from the BOC also, Journal of Bioorganic Chemistry, and you can find out from this the American Chemical Society with this the Sustainable Chemistry and Engineering Journal. From that also you can find out how we can calculate this part. So let us see here the conclusion. We successfully explored a novel as well as the eco-compatible one-pot reactions for the pyrazole dihydropyridine, the pyrimidone derivatives with using the 2-hydroxyethyl acetoacetate and pyridine 2-carboxylic acid and the piperidinium acetate as an ionic liquid. And we maintain that the better agreement with the green chemistry metrics like E factor, atom economy, reaction mass efficiency, effective mass yield, as well as optimal efficiency. And we publish in this bioorganic chemistry journal. So use of a task-specific as well as the recyclable ionic liquid for the development of a novel heterocyclic scaffold. That work we reported in the RSC advances. And some of the compounds shows the outstanding activity against the S. aeruginosa, which is far better than the ampicillin and the chloramphenicol as well as the ciprofloxacin. We reported our work with this bioorganic chemistry journal. Some of the compounds were found active in anti-proliferative screening. And these are the reference and acknowledgement. If I'm talking about this is our Sardar Patel University, and this is our department, chemistry department, UGC Center for Advanced Study. And I special thanks to the Sardar Patel University for providing wonderful opportunity for doing research since last 5 years. And then this is our funding agency, the CRB New Delhi, who funded us for one of the project. And I special thanks to the University Gadjah, Mada, as well as the Aix-Marseille University, France, for their combinations. They provided me a wonderful platform to share our research work. And this is our scholar Diwan Patel, M. Sarma, as well as the Ruturaj Wala, who did this work. The three people they did their work based on the different organic molecules, and we shown here. And these are our collaborators: Professor Joseph Padron from the Spain, Ramesh L. Gardas for the IIT Chennai, and C.D. Anuradha from the Chennai, and Christoph from the Belgium, as well as the Saurav Ganguly from this UK, who help us regarding the microbial activities, I mean based on the anti-proliferation as well as the bacterial activity. So finally, thank you very much for giving this opportunity. So let me try.
Yeah.
So
Thank you very much.
if you have any other question, then you please. I have to stop the sharing.
Yes. Okay, thank you very much, Professor, for the extraordinary lecture. It's very, very clear and has an interesting topic. So for the participants who are curious to ask further regarding Professor Patel's research, please do ask your questions because now is the time. We are opening the Q&A session and discussion session. As usual, as the previous lectures, please ask your questions through the chat box, open your mic, or through the YouTube comment section. Then we are opening the question sessions. Please don't hesitate to ask questions.
Okay, Mbak John, maybe I have a question.
Yes, please, Pak Idham.
Thank you, Professor Patel, for marvelous work. Yeah, very interesting for your presentation. So I have several questions for you. The first one, yeah, maybe we would like to know how to prepare the ionic liquids, yeah, for the solvent. Yeah, and then the second one, yeah, how did you recycle your catalyst in here? For example, the pyridine carboxylic acid.
Okay.
And then if I'm not wrong, when the second talk in the optimization, when you use 5% of the catalyst, you observe the incomplete consumption of the aldehyde. Do you have any reason why?
Okay.
And
so I think in the case of pyridine 2 carboxylic acid.
Yeah.
Right. So, sir, we have used this pyridine 2 carboxylic acid as a catalyst with 5% mole and with 10% mole. So when we run with the 5% mole, then we can have 100% of yield. So not exactly the 10%. I mean, my student will try to do the practical with increasing that percentage of yield. So I mean, increase that the molar concentration of a catalyst, whether any changes is there or not. Otherwise, we know that we have to receive the target as a final product within the 5% mole of this catalyst. Okay, sir. So as far as the preparation of a catalyst is concerned, we prepare the catalyst at a room temperature.
Okay.
As far as the 2-hydroxyethyl ammonium acetate is concerned, we prepare at a room temperature, sir.
Okay. Okay, the next one perhaps, yeah, in the last part, yeah, you show us how to calculate the effective mass yield, yeah, and you show us that there is benign and not benign, yeah. How could we determine which one is benign and not benign?
Please repeat the question. I didn't.
Okay, in the calculation of effective mass yield.
Effective mass yield, okay.
Yeah, you show us that there's a benign chemicals and non-benign chemical. How to determine which one is benign and non-benign?
Okay, let me try to reaction mass efficacy.
Yeah, effective mass yield.
Okay, let me try. Effective mass yield. Okay. So, sir, in the case of effective mass yield, we have taken the mass of the product.
Yeah.
We have to take a mass of the product and mass of the none. That means and the binning reagent. That means that whatever the binning reagent, that means whatever the starting material we have taken, okay, and mass of the none. That means suppose a little bit, if we can have a byproduct or any other some impurities is there, that we have to collect it. And then based on that, we have to deduct it. And then we have to go for the reaction mass efficiency. Sir, for more details, you can see that the brief, the introductions which is given in the effective mass yield calculation on the website, it is available with the supplementary file.
Okay, thank you.
With the Bioorganic Chemistry Journal, sir.
Thank you, Mbak.
Thank you, sir.
There's a question in the chat box from Ahmad Muhammad Yusuf.
Let me try to check. Please, what type of separation used since column chromatography is not used? So suppose if I'm talking about whatever the isomer we have, in this case, I mean most of our product, we didn't use that column chromatography. We didn't use the column chromatography and we have directly the product. But somewhere for example, with the 15 entries, some of the compound we have, so in which we can have some of the impurity. So that can be eliminated So that's why it requires the column chromatography. And we have a automated flash chromatography. Suppose if we found some of the trace amount of the unreactive product we have, then we have to check it through this automated flash chromatography, and we have to remove it directly by taking the fragments. So if you got the answer.
Yeah, I hope it's clear enough. Are there any more questions for Professor Patel? Okay, so I assume there are no more questions, and the presentation on research from Professor Patel is very clear enough. So that is all for today's discussion session with Professor Patel. Once again, many thank you for the lecture and
It's my pleasure.
Yes, thank you very much, Professor, for the new information and for the new knowledge that you've willingly shared with all of us here today. After 3 lectures from 3 remarkable professors, we have finally come to the end of today's session. So before I close today's event, I would like to ask Dr. Idham to give a closing statement. Dr. Idham, the time is yours.
Thank you.
So on behalf of the organizing committee, I would like to thank you, our lecturers today, our wonderful lecturers today. We have Professor Khairuddean from University of Sains, Malaysia, and we also have Professor Diwan Rawat from the University of Delhi, India, and also we have Professor Patel from the Sardar Patel University, India. And also for your participation, it is very interesting. So again, I would like to thank you for your participation. And then for information, our lecture today, you can also access through YouTube. You can find in here, this is the link. Or you can also type this title and then you will find our guest lecture. So I do believe that our lecture today will be useful, and the students or researcher will learn a lot from our lecturers today. So for the information, the next guest lecture, the 3rd guest lecture will be conducted at 12 November and 2021. So we will have Professor Laurent Commeiras and also Professor Grafotto from Italy, and then Professor Gadjah from the Universitas of Mont Blanc in France. So please note the date, the time, and also this is the link for the registration. Okay. Again, thank you for the participation, and see you again in the next guest lecture. Thank you very much.
Thank you.
Thank you, Dr. Idham. Thank you, Joanne. Thank you so much, all the organizer. Okay.
Thank you for your kind words, Dr. Idham. So one last thing, participants, please click on the link sent in the chat box by the committee and fill in the feedback form for us to improve, to be better, and to receive the certificate as well. All right, today's program of Guest Lecture Series World Class Professor Program has really come to an end. Despite of the pandemic, we are all still here and able to gather and learn together. I would like to once again extend my gratitude to all of our distinguished speakers, Professor Melati Khairuddean, thank you, Professor. Professor Diwan S. Rawat, thank you very much. And Professor Hitendra M. Patel, thank you, Professor. And thank you very much for sparing some time to share with all of us to be able to learn directly from world-class professors. Thank you as well to Dr. Idham for organizing today's event and for making it possible for all of us to gather here today. And last but not least, surely we cannot forget all the participants who are eager to learn together here today. So I, Joanne, am personally very grateful to be your MC and moderator for today. Thank you for your active participation, and hopefully one day we may meet directly, and I wish you all to be healthy and happy always. Stay safe, and thank you.
Thank you.
Thank you very much, everyone.
Thank you, everyone.